All right, looks like a little drape of sediment here.
From this angle, I would say more than just a little.
I was being polite to the sediment.
That's fair enough. Fair enough. Let's see. So Del, I'm thinking you were describing how the flat top is a carbonate platform built up by corals, and so might we expect to see in large view what we see in small view in living corals, that is like spurs and grooves and that sort of thing?
Yeah, no, I think that's a good possibility. And of course, as this thing is growing and the volcano is subsiding, we also have fluctuations in sea levels. So there may be times in the past when parts of the reef are subsiding, are exposed, and parts when it's under the water. So we may see all sorts of interesting structures.
The color of the bottom is very different from
Yeah.
these deeper dives that we've been on. Oh, I see a fish already
All right.
sitting down there in a rock, and sea star and a shrimp, and maybe a lobster. So it looks like we're going to see some stuff today. We're just waiting for the ROVs to settle out, the pilots to get settled before we begin asking for various moves and close-ups. Looks like there's a lot of sea stars down here, actually. Bruce Monday says that this is kind of like a box canyon terrain.
Yeah, if you do look over at the sonar view on screen 3 again, you can see some reflections coming from different sides. So yeah, we definitely have some topography kind of on all sides of us here.
And the view in camera 2 in Sirius, you can see that the Deep Discoverer is kind of on the looking at the edge of this one feature that's kind of sticking out a little bit from the wall. This can make it a little bit more challenging for the ROV team as they're flying, and they want to be sure that we don't get the cable hooked up on anything. If you look at camera 2, the Sirius view, you can see this long yellow cable that's coming up from the back of the Deep Discoverer, and that's hooked into the Sirius. So that's the tether that you may hear about now and again. And in addition to connecting the ROV to the rest of the system, it's carrying all the data, the images that you're seeing, and other information on temperature and oxygen that we're reporting and so on. So all the information that we get and the ability to control the ROV and for us to see is moving through that cable.
Yeah, so we're going to be going up a fairly uniform slope here. We're going to go up about 250, 300 meters over a distance of just a little over 400 meters. So we've got a slope of about 40 degrees or so, it looks like. We're going to be climbing on this dive.
Get on your climbing gear. See also what look like a few sea urchins around. Got one fish on shore, they thought perhaps a scorpion fish. Looks like it moved off around the corner. I still see the lobster straight ahead. Shrimp. Maybe a sponge tucked up there up on the upper left. And a brittle star.
Yeah, you can see already we're likely to have more sort of three-dimensional habitat here in the rocks themselves compared to some of the volcanic settings. You can
absolutely good places for octopus to hide.
All right.
Keep our eyes out for that. It's 8.3 degrees Celsius down here, so considerably warmer than our last dive, which was below 2 degrees. And the oxygen continues to fall, 0.57 now. 0.57 milligrams of oxygen per liter. So that's a very low level, and is at the very least getting into, I guess, what we'd call hypoxic or dysoxic
Absolutely.
Yeah. Good morning, pilot. And we're seeing plenty of stuff in the area, so if you think it's time to start some zooms, let me know.
Well, if we can zoom in on the crustacean, it'd be interesting to see what that is, if it's actually a lobster or if it's a large shrimp, prawn.
And we're doing so now, Bruce, so if you think you can identify it. Or Chris, go ahead.
Yeah, I'm pretty sure it's Heterocarpus. I'm not sure which species, so it's a pandalid shrimp. Yeah, maybe Bruce can weigh, or you can weigh in once you get a closer view, but it sure looks like it to me.
I see also a sponge tucked in there and a little crevice above it. Chris?
Yeah, so this is absolutely Heterocarpus. Could even be Heterocarpus levigatus. I can't see the rostrum very well and where it's serrated, but it's got these keels on the thorax and just other aspects of it. I'm quite certain of that genus. So in the family Pandalidae, genus Heterocarpus.
Thank you, Chris.
And that's a genus of commercially important shrimps. They were fished in the Hawaiian Islands intermittently. The populations can't hold up to sustained fishing, so the fishery occurs every few years and then stops. But it's a commercially important shrimp.
How are they harvested, Bruce?
It's done by trapping, using baited traps.
Gotcha. Thanks.
And obviously, a terrain like this, if you tried to fish for the shrimp using traps here, you'd lose a lot of your traps.
I would imagine.
It wouldn't be a good idea.
Yes, seems like it would be a very tricky place to try and fish.
And that's even if it wasn't a protected area. And again, I should emphasize that this is a protected marine national monument, so no fishing.
Right.
Is,
The shrimp is safe.
is this a typical depth range then?
Yes, it is. Maybe Chris can provide more information, but definitely this is the depth range for the fishery and the shrimp.
Pilot, I think that's good. We can see that it does have a serrated rostrum. So when you pull out just on the left side, you're going to see a sponge just a little bit higher than where this shrimp is. Thank you.
Yeah, and this is the depth range. There's a shallower species of Heterocarpus in Hawaii, Heterocarpus ventifer. It's more of a swimmer rather than a squatter like this one. These actually get down to about 1,000 meters in certain places. So 500 meters, they may get up to about 400 or 350 at the shallowest end of their range. But this is smack dab in their range. So yeah, no inconsistencies here.
We're going to have a look at this sponge here, Chris. There's some very interesting brittle stars on the wall. We'll get a look at those next. Seen a couple of different species.
Now the ship's door will be opening for the next 10 minutes. Again, that's the ship's door opening for the next 10 minutes.
Oh boy.
Interesting texture.
Whoa. I'll tell you what, Bruce, if you take the sponge, I'll take one of your fish.
Doesn't sound like a fair exchange for Chris, but yeah, I could definitely identify this as a sponge. How's that?
Well done.
Yeah. I see that Shirley Pomponi is in the chat room, but I don't think she's on the phone yet. Or if she's, I think she's traveling, so I don't think she has the ability to be on the phone today.
Actually, I am on the phone, so I can hear you better than I can see you. So
hi, Shirley.
Yeah, and I was having some challenges getting into on the internet. I can see the sponge right now too, and I have no idea what it is. I'm pretty sure it's a hexactinellus.
Yeah, I would agree, Shirley, and I'm seeing a framework there. It looks like it's a framework at least, so it could be one of the hard sponges. Could be in the family Phoridae. That would probably be my first guess, but it's not quite right for most of the members I'm familiar with in that family. So anyway, yeah, it's I totally agree. Glass sponge, Hexactinellida.
By the way, Michelle Kelly from Niwa, New Zealand, sent in some identifications of sponges that we saw at Cook Island a few days ago. So some really rapid turnaround on photo ID.
That's great. Were there any surprises there, Shirley?
She was able to put some names on things that, and I think to confirm what Chris, for some of those, what Chris has been identifying, and then there were some that we weren't quite sure of, and she put some names on as well. So it's nice to be able to have some names to now all we have to do is remember them when we see them again.
Yeah.
Yeah, the one discrepancy I saw is she went to Bolosoma on this yellow stock sponge in the Cook Islands, and we've actually sampled that, and I've sent the spicules to Henry Reiswig, the spicule pictures, and he may have even accessed the specimen itself. And Henry was not willing to go to that genus. It's in the subfamily Bolosomanae, but he wasn't willing to go all the way to Bolosoma on that yet. He thinks it's a new species and a new genus in Bolosomanae.
Uh-huh.
But other than that, yeah, I agree with Michelle's identifications.
Cool.
Bruce Mundy was remarking in the chat room that we're not seeing any corals here, and on previous dives in the Palmyra area, they had seen quite a lot of deep sea coral. And so possibly we're in an area that is not, the currents aren't quite as strong. We can see there's a lot of sediment dusting the bottom, and so that may be a problem for some of the sessile fauna to get a hold and not be buried before they can grow to an appropriate size. We noticed a couple of sponges here seem to be tucked down into the crevices and caves and cavities and so on. So that's one way of not being buried, but that may affect the corals. So we'll see as we go up slope if that changes.
Yeah, the distribution of corals was very patchy. It was interesting. When we did the submersible dives, we'd go along these sort of what I call box canyons and then ridges, basically equivalent of spur and groove habitats in shallow coral reefs. But on one side of the ridge, there'd be very little, and then we'd get to the upcurrent side, and there'd be high densities of corals almost in a line going along the ridge. So we might see the same thing on this dive.
Scott, is that a coral colony over there on the left side of the ledge there, or is it just a rock?
I think I see what you mean.
Right side of the ledge, I'm very sorry.
Yep.
Right side.
So pilot, just about to go off screen, center screen to the right. There's something that's kind of grayish in color. The fish is right over it now. And I believe that might be a midwater fish. We could have a quick look. I'm guessing it's a bristlemouth.
Sure, looking like a
bristlemouth
bristlemouth to me.
Cyclothony, a very abundant bristlemouth.
Okay, pilot, that's good on the fish. Cyclothony, a very abundant midwater fish.
There's a fan, a coral fan there, if you can see that in the upper center right now.
Yeah, I do see it. Yep. I think Roland may have pointed that out. The one at the lasers first. I'm not sure if there's anything there, but the lasers are on it right now. That's what I was seeing. So there's something there. Question is, what is that something? Oh, I think it's another one of the sponges that we were imaging, but this one's dead and just covered with lots of sand and flocculence. If you can go back down, yeah, that's okay on the sponge. Just right. Oh, I see. That's it's a brittle star arm. I thought it was a worm. Okay, yeah. If you don't mind, if we can get to that coral that's on the ledge.
So if there's time, we have a class here at the IRC, and the students are wondering how deep you are and what you're exploring.
Yeah, so we're at about 490 meters right now, and we're on the southwest slope of the Palmyra Atoll. And so what we're exploring today is this carbonate platform that is formed through time over this, on top of this island. It has been subsiding. And we're going to work our way up today to about 250 meters.
Thank you. And the next question is, what do you hope to find down here? What kind of sea creatures do you hope to find?
Well, what we're about to have a look at here is one of the things that I'm really interested in having a look at, and that is a deep sea coral. And these are a type of animal that swim around after they hatch from their egg as a very small ciliated ball. It's kind of like a bunch of cells with little hairs on them that they use as paddles. And they find a good place to live, they settle down, and then they begin to grow into these large fans and whips and other structures. And what we're looking at here now is this big fan, and you can see on the upper left that dark black base that's kind of like the stem, and you can kind of sort of thinking of this growing just like a tree, except that it's an animal. And so you see all those little fuzzy bits that are sticking off the branches. Those are the feeding structures. They're called polyps or zooids. And so this looks to me like it is a primnoid.
I think this is that biflabellate primnoid.
Yep.
Is that what you think too?
It's absolutely biflabellate, which means there's basically two fans that are oppressed one next to the other. And it is in the family Primnoidae, so absolutely, Chris. And we did make a collection of a coral that had this basic structure on the previous expedition. And I can see there's, up in the upper right, I can see a little red arm of a crab. So there's a chirostylid crab in the branches there, and that's not unusual. There's another one right in the center. This looks like a little hermit crab, top center. It's got striped arms, striped legs. And there's two of them. There's one on the far right. And so one of the things you're seeing, and one of the reasons that we're interested in seeing corals in the deep sea, is that the corals act as habitat for other animals. And in this case, we see crabs. We saw at the bottom there were some brittle stars on it. And so they're providing what we call three-dimensional habitat structure. They grow off the bottom just like a tree, and just like birds and squirrels use trees, we have a lot of crabs and shrimp. And sometimes even the fish are using the corals. We've seen eggs that have been deposited on corals, and a few times in this cruise we've seen octopus eggs on corals. Thank you, pilot. And I think I see perhaps a bubblegum coral in the background, a little bit farther on. Yeah, if that's not too far. Did you see what I mean, the little red branches that were sticking up? Yeah, sorry, pilot. Yeah, you saw that? Great. Wonderful, thank you. There's also a large sea urchin in front of that other primnoid colony in the distance. So as we're zooming in here, we're looking at that reddish coral on the right side. And one thing you can note is that there's a lot less sediment right here than there was on that slope that we first sat on. And so that is probably telling us something about the currents being a little stronger over there than they are on the left. Here you can see that large urchin in the background. Yeah, this, let's see, does this have the appearance of a bubblegum coral or a coralliid? This could be a precious coral. What do you think, Chris? It looks pretty rigid. Yeah, I'm going to call this a member of the family Coralliidae, which is the precious pink corals. And Bruce Mundy was noting at the beginning, and Chris, I believe, that one of the things we're interested in seeing and assessing on these slopes is whether there are communities of these precious corals that are used in the jewelry industry. Basically, their skeletons can be polished and used to make different kinds of jewelry. Thank you, pilot. And I believe that is one of those types of corals. So the oxygen, 0.48,
Whoa.
keeps going down.
A new record, yeah.
Looks like maybe another sponge. So it's interesting that just in that spot we saw 3 or 4 corals, but I don't see any others on this ledge. And it says that there's a lot of fine scale control, spatial control over where we see these sessile communities. Sorry about that, had some other voices in my ear. So while we say that if we have a hard bottom with good currents, that's a good place for corals and sponges to grow, but that's not the only characteristics and factors that determine where they grow, and that was a good example because the whole wall wasn't covered with sponges. If we could get a zoom on the sea star, that would be useful. Thank you. Looks like 2 perhaps of the same kind of sea star, but different color morphs. Can we have the lasers on, please? Thank you. So that's a pretty good sized sea star. Those lasers are 10 centimeters across, so they allow us to gauge the size of things. And you can see that this sea star from arm tip to arm tip is bigger than 10 centimeters. So we're having a good look at this 5-armed sea star. And you can see that this is an echinoderm. It's got tube feet on the bottom, and it's a close relative of the brittle stars, and there was a brittle star right above it. And you can see that they are quite different in their morphology. The brittle star has a very clearly defined central disc and long arms that are very mobile, and it can use those arms to push itself across the bottom, where the sea star is using the tube feet to kind of glide across the bottom. And it might be feeding on something there, not sure.
Yeah, last time we saw one up against a rock like this, it was eating on a crinoid yesterday.
Absolutely.
Yeah.
And in the corner of its arms on the upper right side, you can see behind it, tucked away, there's a little squat lobster. Has these kind of light whitish blue with red striped arms. Thank you, pilot. Yep, so there's a white morph right there. And to me, it has the same general structure, so I would default to say that those are the same species, but one is a lighter color than the other. Looks like, at least in this area, the brittle stars outnumber the sea stars. We were asked what sort of things we're interested in seeing, and of course, Bruce Mundy's probably already talked about this with the class, but a lot of interest in seeing the fish. And we've heard in previous dives that the sediments are a good place for the fish to be found because there's lots of little animals in the sediments that they can feed on. But today we have this additional interesting factor that the oxygen level is so low that that may be a problem for some of the fish. You can see these transparent finger-like structures that are coming out of the holes in the upper side of the sea star. Those aren't tube feet, but I think the name is ampulla. And basically, they're thin-walled, and yet they act kind of like gills, so they allow for oxygen exchange for the sea star. And that feathery-like thing that you see on the bottom left, I believe that is a worm living in a tube, and that's the head end of the worm. It's kind of like a fan that it's using to grab food out of the water column. Polychaete worm. Copy that, Nav. Thank you. Looks like a Siphonophore drifting through, and we saw a Siphonophore a little while ago, I think when Chris or Bruce were talking about the, it was when Chris and Shirley were talking about the sponge, and it was really interesting morphology. Very elongated and very well-developed lobes. So definitely going to want to see that in the video replay. It's not one I recognized. This looks like a sea urchin here that is different from the one that we saw on the wall with the coral. Yes, please. Actually, it might be an anemone. Might have spoke too soon saying it was an urchin. Or now I'm starting to think it might be a cup coral.
We'll see here.
Saying that because I see some whitish stuff through the brown tissue. We're going to solve this mystery
Still,
right now.
Yep, here we go.
So again, you can see those lasers. That's 10 centimeters apart, so that gives you a good sense of the size of this animal that we're about to look at. And it is a cnidarian. It is an anthozoan, so it's related to the jellyfish. And I think I'm going to stick with the sea anemone guess because I don't see any septa. And septa, if it was a cup coral, it would be sitting on a little calcium carbonate skeleton that it builds itself, and we should be able to see through the tissue some of the plates that are inside that skeleton. You can see all the little dots on the tentacles. The tentacles are kind of see-through, and they're spotted and spiked with dots. Those are where nematocysts are, and those are little stinging cells. So if you've ever been unlucky enough to be stung by a jellyfish, well, this is the same sort of thing on the anemone. Thank you, pilot. Looks like maybe there's some really tiny cup corals there. There's two just to the left of that anemone. One of them's got kind of a orange color bands around it. Awfully small. Almost sediment grain size.
Almost. So yeah, speaking of the sediment here, you can see that we have lots of different grain sizes when we zoom in here today, and that's because we've got this material basically shedding off of the coral reef platform that we're on. We've been out in the deep sea, we've seen very uniform sand grains, mostly pelagic things, settling to the bottom. So this will be
one of those Siphonophores again. Sorry.
This will be one of our differences we'll see here as far as what types of sediment are coming into the system today.
So I see here a little ledge, and I'm wondering as we pass these different ledges, we move from a little bit more gentle horizontal slope to something a little bit steeper, if that's where we're going to see things tucked away. So far, it's mainly brittle stars.
Looks pretty bare compared to some others. Is an urchin or an anemone?
Well, that's a good question. Can we get a look? Is it possible to get a look on the far right, that larger one? Yeah.
Yeah.
So I think the closer one here is another anemone for sure. But this may be a different kind of anemone. I'm not sure if someone who recently called in still has their mic open, because I think I'm hearing an echo back. So if you would, on shore, check your microphones, make sure you're on mute unless you are speaking to us. Yeah, so I think this is going to be a Pom-Pom Anemone. That would be an anemone where basically you can't find the mouth. It's tucked in there behind all the tentacles. They also refer to this as deciduous, meaning like trees
Okay.
in the fall that
That's-
drop their leaves, this anemone is able to drop its tentacles, and it also can roll around like tumbleweed.
And what circumstance would it do that?
I imagine if it's being disturbed or if it's accidentally dislodged, say, by an ROV.
Huh. Well, hopefully we won't observe that.
No. That is indeed what it is.
Huh.
I think you may have misinterpreted that. But
A manipulation experiment, I think, is what they call that in biology, right? So,
yeah, so this is a different kind of anemone. Looks like the tissue is a little bit more pigmented. It's harder to see through, and what I'm not seeing are lots of little dots like we saw in the last one. That's a good pilot when video's happy. Top center, I am not seeing it. I see a sea star in the distance. Oh, maybe a crinoid right at the top of the screen now in the center. Oh, there's a fish. Let's check out the fish on the left.
oh, yeah.
I think that might be a crinoid up there.
The fish is not going to cooperate now. There we go.
Semi-cooperative. That was interesting, a little acrobatic
So
move
this
by
is
the
one
brittle
of the.
star.
Oh,
Sorry, Bruce, go ahead.
yeah. This is one of the common types of. Okay. This is one of the common types of eels that we see at these depths. It's Bathychondrus, a congrid eel. I'm getting a little bit of echo here for some reason.
Yeah, I still think there's a microphone that is not muted somewhere. And it may be the last person who called in, because I heard the call-in sound and then I started to hear this echo. So please check your mutes on your phone.
Yeah, a good tip is if you're listening on the phone to make sure you aren't also listening on your computer. That's often where that echo is coming from. So if anyone has the sound on their computer, if you can mute that, I think that'll help.
Bruce, what can you tell us about the depth distribution of these eels? I feel like we've seen them at a lot of different depths.
I'll have to look that up. I'll get back to it in a moment.
Again, you can see these brittle stars are quite common. The one that the eel is in front of looks like it might have a swollen central disc, which means that it's recently been eating. No, we're good to move on the fish. Thank you. Go for it. Oh, yeah. I see a coral up here. So we saw 3 coral colonies earlier, and they were on the very steep wall with no sediment. So this is kind of interesting because now it's on the much flatter part, and obviously there is quite a bit of sediment here.
Right.
Oh, well, there's actually, in the foreground, there's a light brown thing almost at the bottom of the screen now. Yeah, so is that anemone again, or is this now? Yeah, that's the same anemone.
Yeah.
Very diaphanous, very transparent. I see it's got white tips. There's a possibility that could be a corallomorpharian, but too hard to tell. Yeah, so I was just going to say, or I guess I did say, that it'll be interesting to see if this is the same kind of coral or a different kind of coral living on this flatter slope. Looks like it also has an anemone at its base. I'm seeing two different colors. That could mean that it's got two colonies very close to one another, a colony that has different color polyps, or the most likely is that it's being overgrown by a zoanthid. We see that very frequently. Oh, look at behind. I didn't even notice that behind it. Looks like a black coral as well. So this will be a good spot to get some images.
It looks like there's a fish to the right there.
A fish on the, oh yeah, I see that, Astrid, thank you. So pilot, I'm sorry, I realize you're turning. I guess we'll just finish what we're doing. There's a fish that's on the right. Yeah, I mean, they're mobile, so it's usually a good idea to get them first. Yeah, it was sitting on, there he is. So now I guess, Bruce, are you able to ID this, or Astrid? I know you, Astrid, you're not as comfortable with these, quote, shallow water fish. This is still deep sea, of course, but the deep
This
sea.
is a rat tail.
Oh, wow, really?
It's a rat tail fish in the family Macruridae. It's in the genus Sclerotinian. We saw these on the submersible dives with the two bands. And on the body, I don't know what species this is. There are a number of similar species, but you see it's got a very pointed, upturned, almost sturgeon-like snout. That's characteristic of the genus Sclerotinian. It's got long fin rays on the pelvic fins, the fins on the stomach, probably a sensory function. The mouth is underneath the head, so it feeds on small crustaceans and polychaete worms that live in the sediments. So it's a rat tail in the genus Sclerotinian, and it's a different species than we've seen before on these dives. There's also a little whisker under the snout that has a sensory function as well. You see the eyes are fairly large. There might be very dim light downwelling from the sea surface at this depth, but the eyes also would sense bioluminescence from prey or potential predators.
Thanks, Bruce. It's so much more pale than most of the rat tails I'm used to seeing. Okay, I think that's good, pilot. We can go back to those corals now. Ken Sulak from the USGS, who's a fish biologist, was noting that we've said repeatedly how low the oxygen concentration is here in the water. And he says, regarding fish occurrence relative to dissolved oxygen, most active fishes require dissolved oxygen above 3.5 milligrams per liter. So here we're at 0.5, wow. Really quite different. Below 2.5 is lethal for most fishes. But in deep water, there are some fishes specifically adapted to withstand very low dissolved oxygen all the time, mainly by passively hovering, so they're not swimming very much. All right, so we are looking at another primnoid octocoral. So this is a deep sea octocoral, very common family, the Primnoidae, very widely distributed, very diverse, lots of genera, lots of species. And this one is being overgrown by the yellow polyps that you see there, and that is a kind of zoanthid, pretty closely related to the sea anemones that we were looking at before. And you can consider these parasitic. They're overgrowing, and so they're displacing the polyps of the coral that produced the skeleton in the first place. See a couple of brittle stars there and in the arm.
Really large holdfast in this.
Yep. Which suggests it's a pretty good age. The holdfast is that disc-like structure that's basically glued the colony to the substrate, to the rock. You can also see on the right side there, a number of the polyps were gone, exposing the skeleton. And that suggests that something has been feeding on it there. That's good. Let's just get a quick look at this brown one behind before we leave. It's on the upper right, out of frame. There it is. Yep. I think this might be a Heteropathes black coral. This would be our first black coral of the day. The first one that we were just looking at there on the left, that's an octocoral, and if we were to look closely enough at the polyps, it's hard to see in this kind of family, but we'll see in some of the others. They have 8 tentacles, hence the name Octocoralia. The black corals are members of the subclass Hexacoralia. They have 6 tentacles. This one's in a little bit bad shape. It looks like some of the branches have lost their polyps, but there you get a good view of some of the polyps with their 6 tentacles. The darker pink is where the mouth is, entering into the digestive space. It's called the black coral because the skeleton is black. It's very spiny. Looks like maybe an anemone has settled on this one. All right, thank you, pilot. Probably the genus Heteropathes, and I'm judging that by the fact that it's got those wide splayed branches plus another set that are sticking straight out towards us. The details of identification would actually require you to look at a skeleton under a microscope and see the shape, size, and distribution of the spines on the skeleton. Nice view. Looks like a large Siddharth Urchin here, just ahead on the left side. It's going to have very thick spines. Not sure what that thing is on the left, though. The red blob, yeah, let's look at the red blob. Just in case it's a little octopus or something. It's a red blob. So I think it might be a tunicate or maybe a sponge. It's growing around what looks to be a little orange coral or hydrozoan, but the color suggests to me that that's coral. Huh. I'm going to say that that's a tunicate. It's bound up with some algae, so that the algae came from shallow water, and maybe this entire thing tumbled down the slope from shallow water.
That's a possibility.
Thank you. Yeah, that's absolutely a possibility. Thank you, pilot. If you have the time, let's get a quick zoom of the urchin, sure. Yeah, Shirley Pomponi says that it could be a demo sponge that is rolled down from higher up on this slope. So this is a little bit different from some of our other dives, Del. We're on the slope of an island, right?
Yeah.
And not on a seamount that doesn't break the surface.
Yeah, so we were commenting on that about the composition of the sediments. But yeah, the same could be true. We could have some more material coming, organic material coming down from different sources here, I guess.
So this is Siddharth Urchin. You can see that there's a bunch of barnacles that have settled on the skeleton, and I saw also there was a brittle star wrapped around one of the large spines. So this urchin is serving as a home for other animals. Those are little lepatomorph barnacles or gooseneck barnacles. Very colorful urchin. The spines are articulated, so there's muscle associated with those spines, so they can actually move them around.
Huh.
And if you were a diver and sort of, poking at it, it would move the spines towards your fingers.
Fairly intimidating, I'd say.
Yeah. All right, pilot, that's good. Thank you. sorry about that. Number of corals coming up here. We must be on close to a ledge. So maybe if we could image them as we're going by, pilot. Thank you.
Yeah, we can see they're on little exposures of rock here.
Oh, yeah.
Yeah.
Good
And
call, Del.
And we can see some bigger ripples, perhaps a little bit stronger currents maybe on this part of the slope.
So let's bypass that large brown one because that's the same biflabellate primnoid. Exactly. The yellow one and then the one just above it, which I think is another black coral. Here in the foreground, that orange thing you're seeing that's got its arms facing towards the left, that's another kind of sea star called the bracingid. We often see those on these deep sea dives. And it's a sea star that's adapted to collect its food out of the water by raising its arms up, and you can see all those little spines. And they can capture various things and feed on them. So this yellow coral, I'm virtually certain, is going to be a Sclerotinian, which is a stony coral. And that would be different from the octocorals that we just described. We had a close look at, and that one that's in the upper right, kind of brownish gold color, that's an octocoral, the Primnoidae. The Sclerotinian corals are the corals I think most people are familiar with. When you hear the word coral, you're probably picturing a shallow tropical reef that's been built up, and what built up this whole Palmyra Atoll, those would be Sclerotinian corals. Stony because they produce this very hard calcium carbonate skeleton. And that is exactly what we're looking at. You can see a very, very smooth, you've got this layer of thin tissue that's on top of a white skeleton. And you can see the polyps all seem to be facing one way, and I'm trying to remember the genus now. I was going to say Enelopsami, but I can see through the tissue there are these striations in the skeleton, and I don't think that's an Enelopsami character. I just have to remember what it was that was the right genus, but it's definitely a Sclerotinian stony coral. Has polyps in each of the tips of the branches there, the little cups, but the tentacles are presently retracted, so we can't see those. That's good, pilot. If we could look just above it at the bluish one. Yep, this is another kind of black coral. So one of the things that's interesting, so this is a black coral, I think it's in the genus Trissopathes, and I said to you before, if you were to look at the polyps are really tiny on this one, really, really small. They have 6 tentacles, and that makes it a member of the Hexacoralia. Well, so too is that stony coral, the yellow one we just looked at. So these two are more closely related to each other and to sea anemones and to zoanthids than any of them are to that octocoral that we were looking at, which is really interesting because this one also has a protein-based skeleton. You can see it's that black. So instead of being stony, it's much more flexible. It's not exactly the same material that makes up your fingernails and your hair, but it's along those lines, meaning it's a protein-based skeleton instead of a calcium carbonate or rocky-based skeleton. And you can see the black through the tissue very clearly, which gives it the name black coral. Thank you, pilot. Not seeing any associates hanging around in that one. No crabs. Actually, there's a couple of brittle stars in the arms, but no crabs. There's that sea star that we saw earlier. A couple more. So let's see. This is going to be another primnoid coral, the white one, which is another kind of octocoral. Oh, what's the large black thing all the way just off screen to the right? It's very flat. Oh, it's just a shadow. I'm sorry. It's a shadow. My bad. Yeah, so you don't need to worry about the one directly under you, but coming on the left, we'll see another white one. There we go. Oh, let's look at the Siphonophore swimming. This is the 4th time it's passed. It's very elongated, and it's very interesting. It has these very long lobes sticking out the upper part. Yeah, so this is a Siphonophore. It's in its own phylum, but a lot of people compare it to jellyfish because it is a gelatinous animal. So it is a gelatinous animal that's swimming in the water column, but it's not using contractions of the bell, which if you ever see a jellyfish, it's contracting. Instead, you could see the light kind of flickering.
Mm-hmm.
Those were things called comb rows. They're bands of cilia that they use to beat, kind of like the oars of a boat, of a rowboat. So it's the largest animal that is known to use cilia to swim or to locomote. So I think the large colony on the left is one we've looked at already, had the zoanthid overgrowth. So let's look above the lasers. There's a much smaller white one next to the sea star. Thank you. So this is, again, a primnoid octocoral, so the family Primnoidae, and I've already said that that's a very diverse family down here in the deep sea. We totally expect to see it. It goes all the way down to, I think, almost 5,000 meters you can find species. Not this species, but other species in that same family. And I'm not certain if this is just a smaller version of the larger one. That's an arrow worm that you see doing a photobomb there in the foreground, typical pelagic predator. All right, so yeah, so this may be a small version of the large one that we see being overgrown with the yellow zoanthid. And one of the things I'd have to do is compare the photos and look to see, for example, these polyps, are they all pointing in the same direction? Are they pointing down, outwards, up? How many polyps are there in each ring? And other characteristics. To identify it again, we'd need it under a microscope. Thanks. Yeah, let's look at that thing that's kind of on the right. You don't know what thing I'm talking about. It's on the right. It's very small. Could be an anemone, could be a cup coral. You'd have to zoom in. You don't even have to move. I'm telling you, it's really small. It's going to be to the right of that white coral. It's at about 2 o'clock from the white coral.
Can we look at the squat lobster to the left as well? To the left of the coral?
Absolutely, Tim. Got to keep zooming in.
Great, thank you.
Oh, yeah, it's just going off the lower part of the screen. It's between the arms of the ophiuroid. Yeah, there you go. It's right in the middle. Is that a little urchin? Or is that an... Okay.
Hard to tell.
It's weird. It's the spines are so thick at the base. It's really interesting. No, it's not. It's not an urchin. That might be a burrowing sea cucumber. I can see a really large opening in the center. Oh, well, I can't even get that to phylum, but I'm going to say it's a sea cucumber. All right, so now we can have a look at that squat lobster that you were going to zoom in on the left of the coral. That is small. There we go, the little guy in the cavity here. So you just heard the voice of Tim Shank. He's at the Woods Hole Oceanographic Institution, and he studies a lot of the animals that are living associated with corals, including these squat lobsters. And here's one living on the ground, so it's instructive to compare them. So Tim, if you would like to say anything about it, please do.
Yeah, Scott, we haven't seen that many squat lobsters in burrows like this on this dive, and not many on the other dives either on this leg. But I don't really recognize this one as being a munida or emunida. You see, there's a couple of red bands on the claws that stretch outward, which suggests it's a chirostylid. Really long pincers. You can see the red pincers at the end there. Sometimes they have a notch on them. Always look for the notch as a diagnostic character. But yeah, it's definitely looks like a. I really can't tell. I'm sorry. Can't see enough of the head end.
I've seen 3 or 4 of these today, Tim. So maybe we'll get another one in a better position. Thank you, pilot.
Yeah, that'd be great. Thanks.
It looks like he's feeding on the sediments there. You can see his feeding limbs just below his mouth, or manipulating the sediments, picking stuff up. And it's worth noting, so...
Hey guys, it's a family Munidae, not Chirostylide.
Munidae, great. Hi, Mary. That's the voice of Mary.
Yeah, I agree with that. Thanks, Mary.
Hey, he's not feeding, he's cleaning himself.
I thought I saw him picking stuff off the surface of the sediment.
Well, he's doing both.
Okay, fair enough. And it looks like we have a large anemone here. This would be a different one than any of the others that we've seen before. But we have seen this morphology on other dives with this kind of golden yellow polyp body. and the reddish-orange oral disc. It's pretty good size, more than 10 centimeters across at the base. You can see the raised, I'm going to call them lips. We wouldn't call them lips, but that's effectively what it is, these raised bumps around the slit-like mouth. And you can see that the tentacles are transparent, but again, I'm not seeing as clearly all the dots that indicate where the stinging cells are. Thank you, Pilot. It's good when video's happy.
Yeah, those structures coming out from the mouth there are a little different than some of the other anemones.
The lines?
Yeah, the lines.
Yeah, so those are basically the flanks of tissue on either side of the tentacle. So every pair of lines you see should be leading to one of the tentacles.
I see.
And they're extensions that you can kind of imagine them as curtains on the inside of the stomach that separate the stomach up to a bunch of little chambers that may make it easier for digestion.
It's just all relaxed here rather than,
Yep.
Okay.
Go ahead, Neff. Does that sound good?
Yeah.
Yeah.
So if you look in camera 2, you can see the view from Sirius there. Give you a sense of the terrain. We're just going to kind of follow this little ridge up here.
Oh, that's a nice black coral there. This is a new one on the right side. So again, it's a black coral despite the fact that it looks very orange in color. The skeleton will be black, and this is going to be in the genus Leopathes. And we may have seen one of these so far in the expedition. Chris Kelly and I were debating about whether that's what it was. We didn't see one of this color, but we're now in the depth range where we start to see this genus. They're 600 meters up to about 300 meters. And their skeletons, one of the things you'll see about the branches is they're much more smoothly curved instead of kind of straight or angular branching. And some of these have been aged to grow very old, and I should probably check the paper first before I say it, but I'm pretty sure that it is, never mind.
You're going to change your mind
I
now,
retract everything that I just said. This is an octocoral. Huh. So this is an octocoral with red senenchymal tissue. That's the tissue that's between polyps, but the polyps themselves are yellow. Yeah, let's focus on that foreground. Huh. So this has the morphology of what we would call the genus Swiftia. You can see it's almost zigzagging the way the polyps are coming out on the alternate sides. Those are the feeding structures, those tentacles. There's 8 of them. You can see the little bristles on them. They're called pinnules. That's a nice hallmark of the octocorals. I see one of those tentacles is being stuck into the mouth on one of those polyps. And if you look at that one branch on the kind of upper left, you see that there's one polyp that has retracted its tentacles. So that's kind of a nice comparison to what they are when they're kind of in their defensive mode, pulled in, versus in their feeding mode, extended. But this is almost assuredly the genus Swiftia, except I've never seen one that is so large and has so many branches. But the color is right and the pattern of the polyps is right. And it's too bad. I think Chris Kelly is not present right now. Chris has a lot of experience.
Yes, I am.
Oh, you are here. So what do you think, Chris? This looks like Swiftia, but look how big it is. And let's get a look at the crab on the left, please. On the right.
Yeah, Scott, I would agree that this is potentially Swiftia. Looks very similar to something we collected, I think it was in the Northwest Hawaiian Islands in 2015, except the colony morphology is different. But I remember it kind of stumped us for a while, and then you actually looked at this under the scope, and I looked at it under the scope when you were in my lab before you took off. Maybe it was like 4 then that we saw this, 2015, and you could see the pigmented sclerites, and I think that's what we concluded. But this is really spectacular. It's beautiful.
Yeah.
And I don't know if there's value to trying to get or consider this for collection, because I'm not sure it's the same thing, but it could be. So
I've never seen one this large.
Trying to grab some of this.
Yeah, I agree, Chris. I've never seen one this large. So if it is a Swiftia, it's possibly a different species, a species we're unknown with. Really pretty urchin down there as well. And while we look at this, I will be looking at my computer screen. We have a chat room with a number of scientists who are offering observations. It looks like there's about 20 or so scientists logged into the chat room right now, and I'm looking to see if we have a consensus on whether to collect it. And I am seeing collect, and Chris has just weighed in. I'm interested. So, Pilot, if we're in a position, we would like to get a collection of this. Wonderful. And there's another, Asako Matsumoto in Japan.
Hey, Scott, this is Tim. Just to the right of where the camera is now, there's an ophiroid with a disc you can see readily, I think, at the top.
With the good
Be
spines.
good to get a snap zoom on that. And then, does Mary have any idea, was that a Galatheid crab we were looking at, that white crab?
No, that's a true crab. It's in the superfamily Myoidea, but that group has been split up into a number of families, and I'd have to get another look at it to see what it is. It could belong to a family called the Pycidae, can't be sure.
Sorry, Tim. When you asked, we had already pulled off, but when we settle back down, try to get a look at it. And I see Sonia Rowley has just joined, and she also agrees that this would be worth collecting. It's 10 or 11?
It's 10 today, yep.
Thank you.
So we're just at 465 meters today.
So it's interesting, if we do see a Leopathes later, you'll see a lot of similarities from a gross scale to what we're looking at here.
I'm just looking. It looks like great habitat. It was full of associates there.
Yeah, it's got a lot of associates. There was some interest in the brittle star. Pilot, whatever is easiest for you to collect, I don't think you're going to get something that's too small. So thanks. And this should cut fairly easily. It's got a thin protein skeleton. But it will not tug very easily, so I do think you have to cut it. Great. Yep. I'm not sure you're in the cutter. There you go. Yep. You got the main branch. I think you might just have a little touch of the other one. You can see how tough that protein-based skeleton is, and that's why we're suggesting cutting it. There you go.
There we go.
Now you got another piece. There you go. Great. No, I think just get into box. And I can see that it is a little bit light. I'll watch as it goes into box to see if there's any ophiuroids or anything in the branches. Oh, I see a lot of warm water coming out of that box right now. Could we just hang on a second while that empties? Yeah. So one of the things that we have found in working with deep sea animals is that, interestingly enough, temperature is much more important than pressure when you're looking at the difference of them moving from the deep sea to shallow water. So for many organisms, you don't have to actually maintain the high pressure, but you do have to maintain the low temperature for them to come up in good condition.
Yeah, so when we're sampling here, when things come up, the first thing we do is put them in some cold seawater and keep them there until we've preserved them. So,
yep.
Yeah.
Plunge our hands in the cold water to get them out of the bio box.
Suffering for science here.
Yep. That looks good. Thank you, pilot. So it's probably worth noting here that this is a NOAA ship Okeanos Explorer, and we are on a mission of exploration, and we try to target areas of the ocean that have never been explored before. And today we're diving in the Pacific Remote Islands Marine National Monument, the Kingman Palmyra unit, and we're on the slope of Palmyra Atoll, diving in an area that hasn't been viewed, and we're just looking at this coral, and none of us on shore recognize it. We have a sense of the genus that is in. You've heard us say the words Swiftia, so we think it's related to a Swiftia, but it's a much larger colony than any of us are used to seeing. And so this is one of the things that we do during exploration. We're looking for the unexpected or the unknown, trying to shed some light onto what may be within the resources of the monument, things that we didn't even realize that we were protecting, perhaps. And we often have, we're lucky enough to find a lot of just amazing behaviors as well. And so hopefully today we'll see something interesting in terms of predation event or feeding or who knows what.
Right. Yeah, you can see this is just a great way to sample. We have a lot of context about where the sample came from. You can see we just took a little bit of the coral. We didn't really destroy it in any way. So.
Absolutely, and so it's so different from sampling with nets or trawls or dredges because, as you just said, the context is so important. So we now know what kind of bottom it's sitting on. We know these various organisms that are associated with it living in the branches and so on. Having the ability to have this wonderful ROV video, high definition, makes a big, big difference.
Yeah. Yeah, just looking at this view, you can see where that little bit of rock outcrops and all the life, most of the life is kind of along that strip there.
Pilot, before you leave, there's somebody who wanted to see something else in that colony. Thank you. All right, Tim, we're going to go back in and have a look and see if we can see the ophiuroid that you were pointing out. Just briefly, want to make some headway here, and hopefully we'll still see the central disc.
So I'll take a stab at this sea star, Scott, since I guess Chris Ma hasn't been checking in. And if I'm wrong, that usually prompts him to call in because it's fun to correct my IDs. But that could be Mediaster ornatus or something like that, the one that was on the left of the colony there. And I think there's, at least we had recorded what we thought was Mediaster back in 2005 on dives in Palmyra and Kingman. So, and Chris had weighed in on those images too.
Thank you, Chris, and thank you, Pilot. We're ready to go.
Thank you, Scott. I don't think we've seen that ophiuroid morph on this expedition.
I don't believe we've seen it on a coral. I can certainly say that. If we have seen it.
The disc was pretty unique there, yeah.
It is something that would have been on sediment, so good eye, Tim. Another one of the Pom-Pom Anemones here. One that sheds its tentacles. I think it's in the family Liponemidae, because I know people on shore are looking for more Latin names.
Get Latin credit if you tune in here.
Almost.
Yeah.
Looks like there's a coral out on the edge of this wall, this feature that we're following to the north. Our depth is 465 meters. It's 8.48, 8.5 degrees Celsius, so that's pretty warm for deep water. We're still in extremely low oxygen, although at least now it's going up, so it's at 0.6 milligrams per liter. Get a good sense of the drop-off here, and if you check out camera 2, you'll see the view from Sirius. You can see we're in some fairly steep topography. So big changes in, I don't know that these are spur and grooves or erosion channels or whatever, but you've got some flat spaces and some steep walls. Thank you. So we have seen this before, really interesting, almost exactly what we were seeing earlier, that we have this zoanthid, this kind of parasitic colonial hexacoral, the yellow stuff, overgrowing this primnoid. There's a beautiful...
wow.
It's related to a basket star, I believe. I don't recognize that. Sorry, Pilot, I'll try to make words. We're looking at the brittle star there on the lower left, and that is not one I recognize. And I was going to suggest that it was a member of the Astroscumatidae, and that's the family that includes the basket stars. But Tim, what do you think?
Yes, Scott. We haven't seen this one either, and it looks more like an astroporpa, something like that.
Cool.
It's worthwhile to get some good video of this, but because it is different. It's a different genus, maybe even family. Start looking it up.
Thanks, Tim. So it's on a primnoid octocoral. Family Primnoidae, and you can see it's being overgrown by the zoanthids. We've now seen this several times in the dive. The question of interest will be, do these zoanthids associate with any coral, or are they only associated with the primnoid corals? So everybody on shore, that's one of the things that we'll be checking out for today. That could be a good question. And I see a little hermit crab there, also working on the upper left. That's good for us, pilot. Thank you.
So Scott, for this primnoid coral and that other one you imaged a little while ago, I would suggest the genus Narella on this, and it could even be something like Narella alata, because it looks like a lot of the branches are coming out near the base, and that's one of the characteristics of Narella alata and Narella musicae. But I think it's reasonably safe to go to Narella as a genus. What do you think?
Yeah, I think that's a good, safe bet. It's a very common and speciose genus, and it certainly has some of the characteristics of Narella. And Chris, while you're still there, are you seeing the urchin that we're looking at?
Yes, I am, and I think this could potentially, it's a little bit of a weird view because it's so close up. I'm used to seeing these from further away, believe it or not. But I think this could be that weird podinid, Cenopodyna,
right.
That's on the walls here. And I think I also saw an Enelopsamia that yellow one up above that as well,
yeah.
The hard coral, but maybe you've already seen that already.
Yeah, we had a look earlier at a colony that was like this, and I thought it was similar to Enelopsamia. However, the skeleton seems striated. And I thought Diva Aman had said earlier in the leg when we saw something like that, that there was a related genus, not Enelopsamia, but something else, and I don't remember the name. But certainly that is a stony coral, branched. Enelopsamia is a good guess. Again, all the polyps have been retracted. Looks like there's maybe an anemone sitting on top of it. You see the one thing that has the tentacles hanging down. Thank you, pilot. We can move.
Yeah, again, the sort of average slope here over our whole transect is about 40 degrees, but locally we've been seeing much steeper surfaces than that.
You can really see all of the architecture and the pitting from the corals and all the various bioeroders that live in coral reefs. Lots of clams and worms and sponges and various other kinds of invertebrates that are digging into the coral, typically to find a hiding place to live, not so much that they're feeding on the coral skeleton.
Yeah, very different in the sort of hard rock, manganese-crusted places we've been diving on many of these.
So this is interesting. So earlier we saw one of these brittle stars and it had 4 arms, and I just assumed one of the arms had been broken off. Here's another 4-armed one, but it does look, yeah, I guess maybe that's a newly regrowing arm there, or is it feeding on something? That white spot just on the top of it. Huh.
Wild.
Yeah, I'm going to guess that that's a new arm beginning to grow. That's good. Thank you, pilot. So, Del, one of the things I know that folks have asked you, and so it's a bit of a leading question because I kind of know the answer, but you've been asked repeatedly about rock samples from these carbonate platforms, and I know what your answer is going to be, so maybe we can tell me a little bit about, or tell the audience a little bit about why that is or is not a good idea.
Yeah,
useful.
we've been trying to collect rock samples on many of the volcanic structures to look at their geochemistry and to look at the age of these seamounts. Now, people do date corals and look at the geochemistry of corals mostly to reproduce paleoceanographic conditions. But those samples are really most useful if we have a kind of continuous core. We can see how things are changing through time. So if we see something particularly interesting, we might try to grab it. But a loose piece of coral rubble without kind of a longer context is probably not all that exciting to most people. So. We don't expect we'll take a sample, but sometimes we get surprised if we see something very interesting. We might grab it.
Great.
Yeah, as we're moving up here in the Palmyra and then up to Kingman next, this is really just a large carbonate platform. And so this is very different. We're going to have this source of carbonate, and we can see if we zoom back out in the topography here, and we'll look at some of this in our call probably even tomorrow. We can see these channels and see these islands, these reef complexes are really a source of sediment that's getting channeled down into the deep basin, into the abyss. So again, very different than what we've seen.
I'm noticing the sediments here on the right kind of like are pocked. And at first I was going to say, oh, look, they're rippled, and it's not rippled. And I imagine it's a lot of reworking of sediments by fish and crabs and shrimp and maybe worms. And I haven't seen any sea cucumbers, but who knows?
Mm-hmm.
Is this a fish or a shrimp drifting? That is a Siphonophore. So Siphonophore is a colonial cnidarian. It's a hydrozoan, and what you're seeing there now on the right side look like a whole bunch of bells that are squeezing, and those are the swimming zooids. And at the top center, there's something that's white. That's a little buoyant structure, so it's got a little bit of gas in there, give it some buoyancy, some flotation. And then the stuff that's hanging down below that, that would be your feeding polyps. So they would have tentacles and stinging cells and so on. So a Siphonophore. Another large primnoid fan right on the side of that rock between that channel, so it's probably a good place to be.
Mm-hmm.
First of all, it's elevated from the sediments, and then it's also in that channel. Maybe the water gets squeezed through there a bit.
Yeah, you can see right around that outcrop there, all the sediments are basically scoured out. So yeah, we can imagine things getting kind of funneled around there.
Yeah, another good indication of fast-moving currents. There's a large anemone. On that rock on the left was a large brown-colored primnoid, and this white one is a white-colored primnoid.
Oh,
fish. So I think we saw this right at the beginning of the dive and weren't able to image it, so this is a good observation. Stand by for our fish colleagues on shore.
Yeah, this is a scorpion fish. I can't identify what species. Astrid had suggested that the one that we saw before might have been something called Sitarches, but I don't think so. It has a rather large head relative to the body, which is characteristic of the genus Pontinus, but this doesn't look like the Pontinus that we usually see. They have a different color pattern, and they also have little whisker-like objects over the top of the eye. The scorpionfish I find very difficult, almost impossible to identify, because a lot of the characters that you start off with are things like the teeth, the nature of the teeth inside of the mouth, on the roof of the mouth, and things that we can't see easily. I'll have to spend some time sending images to some of the experts on the group to figure this out. But it's a scorpionfish. They're called that because the spines on the fin on the back, the dorsal fin, have venom glands and can sting. So that's why they get the common name.
Great. Thank you, Bruce. Thank you, Pilot. Interesting. I was scouring the rocks there for all kinds of little stuff, as you know I do, even when they're on the
So
lab.
maybe we should collect a sample so we can get some smaller scale biology up here. That's fine.
That's a really good point.
We
I
can
hadn't
do
thought
that.
of I hadn't thought of that at all.
As far as numbers of things, we may have got a larger number of things off the rock samples than we have collected independently. So.
No, I think we're pretty good. We can push on. We've seen all of these things earlier. These are two different kinds of primnoid colonies, the brown one and the white one. There's another one of the brown ones. With the sea stars. Those sea stars have been not uncommon so far today. Again, this really thick draping of sediments, and you'll notice that these corals are always going to be on a little bit of a high point because they need to be attached to a hard substrate, and they can't just grow out of the sand. They don't have the proper base. They can't anchor themselves. There are other sessile organisms that do. We just haven't seen them today, but things like sea pens. There's maybe something new, lower center, just going off screen. It's kind of grayish spike ball. All right. Roland thinks that might be another Pom-Pom Anemone. The color is a little bit different.
It's because this one is cheering for a different team, Scott.
Oh, jeez. You're on form, Chris. Very well done. Oh, yeah. Oh, absolutely. Yeah, that's the same anemone, so we don't even have to go in on the anemone. Looks like another one of the scorpion fish we just imaged. Yep.
Yep, so this is another scorpionfish, and it looks like a different species. It seems that the color pattern is a little bit different, although the head shape's the same. Scorpionfish can be variable in color, so I'm not really sure if this is a different one or not.
Thanks, Bruce.
Yeah, oxygen's only 0.65 milligrams per liter, so seems not to be a problem for the scorpionfish.
It's a good thing he's just sitting there. Looked like there was a little yellow jellyfish that sort of moved off the bottom just behind that fish. That was kind of neat. The coral fan that's on the upper left, that looks different from anything we've seen today. Thank you. I did a dive in one of the Hurl submersibles, the Pisces 5, and we were on Penguin Bank, which is a large carbonate bank off of Lanai in Hawaii. And this reminds me of it a lot. We were quite a bit deeper, 1,400 meters, but the same sort of structure to the rock and ledges that had sediments on it and then free vertical faces. Oh, yeah. So this, I believe, so this is definitely another octocoral. You can see there the skeleton has been exposed as kind of brownish in color. There's some tissue that's been missing. But this would be in the genus Acanthogorgia. And one of the ways that I can tell that is how skinny and tall the polyps are, relative especially to how thin the tissue covering the skeleton is. And there would be other characteristics. If we were to be able to see it close enough, or if we had it in the lab, you would probably see kind of a crown of thorns up around where those tentacles are. And there's a sea anemone that has kind of glued itself to the branches. It's pointing away from us. Oh yeah, if you look at, oh, shrimp, there's a call them a school of shrimp?
I'm sure there's a word.
I'm sure there's a word. There's a whole lot of shrimp that you can see in camera 2 hovering around the umbilical. So
Yeah, the question is, are they shrimp? Are they shrimp or krill or mysids? They don't really look like the Naphophasia mysids, the big red ones, but we did collect those with midwater trawls here at Palmyra.
I just want to point out the science. If they look in the center, just
They're
past
not
the coral, there's some
Naphophasia.
solitary hydroids. I've seen 2 or 3 in the imagery.
The word is swarm.
Swarm, thank you. All right, another urchin here.
They might be suggested shrimp. Family called it suggestidae.
Yeah, so this is a Narella, genus Narella, a primnoid arctocoral. And the question that we have to answer, and I probably won't be able to do until the end of the dive when I'm comparing different images, is whether this is the same species as the one that we're seeing overgrown by the zoanthid, that yellow polyp structure. Those ones were all bigger, and so maybe it's only when it gets older, zoanthids can settle on the branches, that it becomes overgrown, and at this younger stage, they're still free of that sort of parasitism. I don't know that that's the case, but it could be. And I think that is, yeah, that's another little hermit crab on the far right there with that snail shell. I can see one of its claws extending in its 2 little eyes. It's a little one. Thank you, pilot.
Could we take a look at this red thing on the wall that's to the right?
We are headed that way, Chris. From this angle, it's looking like another octocoral. Yes, indeed. So this is what we were seeing earlier. I was trying to decide if it was a coralliid or a bubblegum coral. And this one looks more like a bubblegum coral to me. You can see the lumps at the end of branches sort of take on that appearance of a wad of bubblegum, and you can see the polyps that are extended. So Chris, what do you think? I think it's Paragorgia.
Yeah, I completely agree, Scott. Sure looks like a bubblegum coral to me.
Great, thank you. So one of the things that can be a little bit tricky is separating the bubblegum corals from the precious pink corals. Sometimes, oh wait, let's have a look at the lower part of that colony. There's a large sea slug making its way. It might be feeding on that coral. And this is not a sea slug that I recognize. Cool. I can't tell if that is a nudibranch or if it's some other kind of slug, but you know, they're all gastropods, so related to the snails. Yeah, that'll be good, only if it doesn't take too much time, but great, thanks. Chris, you know, I was just describing how we're trying to separate out the bubblegum corals from the coralliid corals, because it's the coralliids that are the precious corals that are subject to a fishery. The bubblegum corals aren't. And if you look at the skeleton where the tissue has been removed, I see striations in the skeleton. That is not something that I would expect to see in a bubblegum coral.
Ah.
Doesn't mean it isn't, but it's not what I would expect to see.
I'll be going to a malacology meeting next month so I can take a picture of that creature with me.
Great. Thank you, Mary. That's Mary Wixon at Texas A&M University.
Yeah, I do see the striations there, though.
Yeah. So I can see those structures are called rhinophores. They look like two little ears at the forward end of that slug. Those are sensory in nature, and then you can see the kind of the frilliness on the top side and then the muscular foot. And so it's not actually on the coral, or is it? I guess. I think it's. Yeah, I think it's on the coral because I think I see pink through the tissue. All right, that's a great view of both that skeleton and of the slug. So thank you, pilot.
There's also solitary hydroids in the back on the substrate.
I'm going to get a good look at those one of these times, Roland. I wanted to say that I've been getting some help from Angel Valdez, who's at Cal State University, on identifying the sea slugs and the nudibranchs that we're seeing. So he's at Cal State Polytechnic in Pomona. It's like a larger version of that yellow Enelopsamia stony coral. Yeah. What's the white thing that's right in the center, just above the laser? All right, let's follow our curiosity. Yep, understood. It looks like a bivalve. Oh, no. Oh, good call. Someone said sea urchin cell. This is a Spatangoid urchin, an irregular urchin, known as a heart urchin. And so this would have very short spines, and usually they're burrowing in the sediment, so you don't often see them on top of the sediment. This one's dead. You can see that the shell has been broken open. So basically a relative of the sea urchins. Thank you. You see a couple more of these yellow stony coral colonies on the right side, raised up on the elevated part of the carbonate, and then another one of the primnoids. Looks like a brittle star. I'll say yet another different one. I don't think that's the one we saw on the primnoid colony. It's a really beautiful color. So you can see two brittle stars next to one another. Big color variation, and also variation in the length of the spines that are on the arm. Also, how big the central disc is. So a number of differences there. And this is really interesting. So actually, if we could look at this a little bit closer, above the brittle star were all these, I thought it was mucus. There's a whole bunch of very fine tentacles, and they're leading back into something red that's probably in a tube. And I suspect that that's a polychaete worm. There are things called spaghetti worms and spaghetti mouth worms. They have lots of tentacles that they kind of lay over the bottom, release a lot of mucus. Particles get stuck in it, and then they can kind of be carried back like a fishing line to the mouth. So I suspect that's what's in there. It's a little bit hard to see, that's for sure. Yeah. That's good, though. That's good, because we often see what we refer to as a spider web down here, and so maybe it's something like these polychaetes that are causing it when they lay all those tentacles out and they emit mucus. Thank you. While we're there, if you zoom back in, there's a shrimp on the above the laser, above the left laser. Since we have Mary Wixton on the line and Tim Schenk, very interested in all things decapod. Really beautiful shrimp. Large eyes on stalks. You can see the base of the antennae have a number of bristles, seedy, extending from them. And they would be very effective at detecting molecules in water, so basically sniffing the water. You can see the beating of those little structures under the eyes. Those are legs that are associated with the mouth, modified limbs that are used as mouth parts for manipulating food, cleaning off the antennae, and so on. Great, thank you. Nice view.
Might be a genus called Heterocarpus. I can't see the ridges on the carapace that will be characteristic, but it does have the striping we've seen on Heterocarpus.
Great, thanks, Mary. So we did see a Heterocarpus earlier in the dive from a couple of different angles, had a very prominent rostrum, serrated rostrum, and was identified, but I don't think it was the same as this one. It was much more robust.
Yeah, Mary, I was wondering whether that could have been a Plesionica, just a head-on view of one, but I wasn't sure. That was an odd perspective.
Yeah, the perspective's hard. I didn't see the carini that you would expect in Heterocarpus, so it could indeed be a Plesionica. At any rate, it's a family called the Pandalidae.
So we see some line here on the bottom. We just want to make sure that it's not extending up into the water column and no chance of it getting hung up on the ROV.
So even though Palmyra is protected now, this is an area where fishing occurred in the past. There was even a long line vessel that ran aground at Palmyra a number of decades ago. So we're probably looking at remnants of fishing gear from the past before the island was given protection.
I guess another possibility is, given your location right outside of the channel, maybe somebody tried to anchor outside of the channel and their anchor got stuck. I can't see if there's any rope going into some hole where there might be an anchor, but
we
yeah, who knows?
We can see one end of that line, Chris. Looks like it's free, not attached to anything, but we haven't found the other end of the line yet. There we go. It's hard to tell if that's like cable or line. See a lot of these very tiny white stalks, little pedestals or thumb-like structures, and they're so tiny that around that sea star we just had a close-up of, there were probably 10 of them. And we did collect a couple of those off the rock, one of the rock samples we got earlier in the expedition, and Chris Kelly was telling me that I believe they collected one on maybe in 2016. And they may be little tiny hydrozoans. It's just amazing how small some of the life gets here and tucked into nooks and crannies and buried in the sediment. And all of it is providing food for the larger animals that we are seeing more easily. And when I say large, we can image things down to just a couple of centimeters. So we do see small stuff, but there's even smaller stuff that we don't see. Wishful thinking. You can imagine all of the things I'm seeing in these rocks and holes. Lots of urchins feeding around that line. It's kind of interesting. I wonder what it is about that little spot. Our depth is 448 meters. Temperature of the water is 8.3 degrees Celsius. Very low oxygen concentration still, only 0.6 milligrams per liter. And we heard earlier Ken Sulak, fisheries biologist at USGS, noting that most of the active fish would not be present below 3.5 milligrams per liter of oxygen. So this is really quite low. We're diving on the slope of Palmyra Atoll, and we're in the Pacific Remote Islands Marine National Monument in the Palmyra unit. Some interesting larger rock here. It's too bad Dell has just gone to lunch. Bad timing. This looks like it's tumbled down from somewhere above, and it's got a coral perched on the upper side. The coral's found a really nice high spot in which to grab food from the water column. So if we're going that direction, we'll get a look at that. I'm not seeing anything. This is a Pom-Pom Anemone in the lower left. We've had a look at a few of those. I see more urchins and sea stars. Another yellow stony coral.
There's a fish that's coming right down by that ledge.
Yep, that's what we're trying to get.
I think the pilot spotted it already.
I'm looking at that motoring sea urchin, how fast that's moving. Oh, little triggerfish, I think?
Well, this is a spikefish. It's a triggerfish
right.
relative. It's in the genus Holardia, similar to or the same as Holardia goslinii in the Hawaiian Islands. And both of the genus and species names are after scientists. Hollard was a early, I think, 19th century naturalist, and then William Gosling was the ichthyologist at the University of Hawaii in the 1950s. So it's a relative of the triggerfish, the filefish, and even the puffers, Holardia.
Thank you, Bruce. I think that's good on the fish. And we also saw, I'm not sure, Bruce, if it was the same species, but we definitely saw the Holardia. I think it was at either Jarvis Island or that small cone that was also in the Jarvis Monument area.
You know, the interesting thing is that we've seen Holardia at most of the general locations where we've done Okeanos dives here in the Pacific at these very shallow depths on the shallowest dives. And until the Pearl dives in the Line Islands and then the Okeanos work, these Holardia were only known in the Central Pacific from the Hawaiian Islands, so they're much more widespread than previously thought.
Great. Looks like an Oreo down here, Bruce.
You see the Oreo, Bruce?
You know, this is the Neophytus species, similar to the Neophytus acanthorhynchus of the western Indian Ocean. It's a zeaform fish. It's a dory, Oreo dory. These are quite abundant, and even more remarkable than the Holardia, these were totally unknown from the Central Pacific Islands before the Hurl dives and the Okeanos Explorer expeditions.
So again, we get back to this theme that I'd been mentioning earlier about the value of the exploration, revealing things that we hadn't realized were there, because we're able to get down here basically with our eyes and get some really good views in some of this complex habitat, complex topography that's really hard to sample in other ways. Science is good when video is good. Oh, really large whelk down there, or some kind of predatory snail. You can see that it's probably about 20 centimeters in length. I'm not not sure. It's it's kind of a weird shape, isn't it?
It looks like a shallow water Triton's trumpet shell, genus Coronia. And those feed on urchins. We saw that test of a heart urchin earlier. However, this shell looks dead, so it's probably something tumbled down from more shallow water. I don't know. They may be around. Just a guess, though. I'm not a mollusk expert. I just recognize the Triton's trumpet from diving in shallow waters.
Great. That's good information, Bruce. Thank you. Yeah, and you can see it's in a pile of other debris, so maybe all of that is tumbled down together. I couldn't see the foot, so there's no indication initially that it's alive. Yeah, you can see down on the right at the bottom of the screen, just to right of center, the opening into the shell, the aperture, and there's nothing in there. You can also see what's really instructive. There's a black dot to the right. And that, the edges of it, what you can see, that's probably an oyster drill or some other snail has actually drilled that hole and then probably fed on that snail from the outside. There's a variety of different snails that can do that, and they use their rasping radula along with secretions of acid to gradually drill those little holes. They also do it in oysters and other kinds of bivalves and so on. And it's possible that there's a little piece of black coral skeleton. You can see that black thing that's really spiny. That's my guess at what it is. All kinds of stuff here. All right, great, thank you. Yep, it was right on the top on the leftish side. See a variety of urchins on this rock. There's also something red. What was it red that we zoomed in before? Oh yeah, it was a bubblegum coral that had those striations though that make me wonder if that was actually a correct identification. Lots of urchins on the top of this rock and on the side. We saw this also on the slope of Jarvis Island, so we're seeing some similarities in communities between these two places. Jarvis is just a little bit further south, well, actually about 300 miles, I think, further south. It's just below the equator. And we were a bit...
Yeah, so these are the strange pedinid urchins, according to Rich Mui at the California Academy of Sciences, and he went to the genus Cynopodina. But we've seen some of the Cynopodina elsewhere, and as you may or may not recall, they're the pretty red and green spined urchins. So obviously these look quite a bit different than that. So I gather this genus can have a variety of morphologies.
Thank you, Chris. That's Chris Kelly at the University of Hawaii. The colony that we're looking at here, I believe, is another Acanthogorgia. So we saw one of these earlier. Acanthogorgia, the name translates to spiny gorgon, spiny coral. The term gorgonia is used for essentially all of the sea fans. And this one also has one branch that has been parasitized and overgrown by this yellow zoanthid. Completely different species. It's actually one of the hexacorals, so it's more closely related to the sea anemones than it is to the coral that it's growing on. And then you can see in the arms is what we call an astroschematid brittle star. That's the family. And it has these characteristic, pretty thick arms, very well wrapped around and entwined with corals, and we see these very frequently living on corals. And then I can see a little squat lobster tucked away just at the base of that yellow branch. Is kind of hiding behind the white polyps. And if Mary's still in the line, if we can get a better view, maybe she can give some comments on that squat lobster. Dell's back. I'm about to go to lunch. And Dell, what we're looking at are a couple of massive blocks that seem to have dislodged and tumbled down. So very different from the surroundings. Okay. Great. Beautiful, beautiful image. I can't leave right now. This is just too nice to look at. You can see the zoanthid polyps. They're really rough. The polyp body wall, the column, is really rough. And they are able to embed sand grains and other debris into that wall. And that's one of the reasons that it may be rough. It may also be some of the production of the type of cells, stinging cells and so on, that we're seeing a reflection of. Or it may even be parts of the coral that is growing over. again, the Acanthogorgia, you see those polyps are really tall and slender relative to the thickness of the tissue that they kind of crunch up into. And I would have liked to have seen some of these polyps retracted. And you might be able to better see the sort of pin cushion in that way. Let's have a look at this urchin on the right, see if he's feeding on the coral or if it's just next to the coral. Oh yeah, these are the podinids, sorry. I already forgot. It was 1 minute ago. Definitely time for lunch. All right, Del, it's up to you.
All right. Biologists, don't be shy if you see something you like here. You can.
We've dropped the call. I'm calling back in.
You have reached Verizon Conferencing. The number you have dialed is not currently in use. If you are trying to join a meeting, please verify your information and try again, or contact your meeting facilitator or reservations department for assistance.
I think the call's dropped. Yeah, so if you're watching a live feed or you're on the tele line, I think we've dropped off the tele line, and we're trying to get back on.
There are 5 parties.
All right, can folks on the teleconference line hear me now?
Can do.
All right, great.
Nope.
Got disconnected there briefly. We're back. Yeah, it looks like the rock to the left here is definitely slid down slope, but what we're seeing here on the right seems to be in place. So if you're just tuning in today, you're watching live video from NOAA's ship, the Okeanos Explorer, and we're diving today on the southwest slope of the Palmyra Atoll in the Central Pacific. We're about 360 miles north of the east equator at 162 degrees west longitude. I'm just zooming in here to have a look at this coral. And we certainly have seen this already today, I think. I don't see any, trying to see any associates in there. Maybe, I don't think so. I think science is good. Yeah. It should be. Yep. Yeah, we can go ahead and take a look at this here. If we can zoom in. Looks like a sponge down at the bottom.
Oh yeah, I'm waiting for that sponge. So I'll take a stab at this. This could have been what we saw earlier. This is a little bit better view of the sponge that neither Shirley nor I could do better than hexactinellate or glass sponge for the ID. But what you've got down here in this area of the Pacific is a genus called Alloplax, and it's in the family Alloplacidae. And one of these was actually collected. I'm not sure it was Palmyra. It might have been at Kingman or Jarvis. I can't remember right off the top of my head. And it was identified by Henry Ricewig to that particular species. I'd never seen it before. And so that was really cool. And they have a very distinctive kind of ribbing, as you can see, your channelization and they form sort of these cups as well. So I think this is what we're looking at. I think we're looking at a glass sponge in the family Alloplacidae and in the genus Alloplax.
Great, thanks, Chris. Let's look up here then. I think science is good with this. So we can look at this whiter stuff just above it. Looks like there's a bunch of this. Looks like something dead here, I guess. But is this another sponge, or is this a piece of a face of an old coral? What are we looking at here?
I think you're looking at old coral, Del.
Yeah.
That would be my guess.
Looks like that maybe a little cup coral there growing on that. Yeah. Right, I think science is good. See another one of those yellow stony corals. And I know we've seen this red coral as well before. I think we've seen everything else here. Is there anything else anyone wants to zoom on? Okay. All right, we can head on up then. Yeah.
Well, I couldn't recognize that fish. It was too close to the camera. It looked like it might have been a small midwater fish, maybe in the family Sternoptychidae, one of the more slender-bodied species in that family. But really have no idea.
All right, thanks, Bruce. So this structure here looks a little familiar from what we saw earlier. Someone want to offer an ID? Yeah, just curious here. It doesn't.
Yeah, Del, it's a black coral for sure, and boy, better than that, it may be Piranopathes, but I really don't know. Actually, the bottom branches are all denuded of tissue, so the top part is the live part of it, but looking at the branches on the bottom is also informative, and still can't figure it out, but my best guess would be Piranopathes.
Okay, yeah, I think we might have seen an example of this earlier. I remember that sort of radial pattern there of the branches. I'm guessing this would usually be attached to rock, so it looks like it's going into the sediments here, but maybe it's just a small bit covering up the holdfast. I don't. All right, Pilot. I think.
Yeah, I'd agree, Del. I don't think these things. This is not one of the black corals that actually can anchor in the sediment, at least to my knowledge. But I could be wrong.
Great, thanks, Chris. Yeah, I think science is good. Yes, this is to me like the same coral. I forget, what did we decide on? Is this a precious coral or was this the bubble gum?
So Del, I suspect that this is going to be a little bit different species of bubblegum coral, or maybe it could be the same species, but all the polyps are out on this one, whereas the other one that we took a close-up of had all the polyps retracted. Yeah, I think this is in the family Paragorgiidae and possibly in the genus Paragorgia. It's a guess, but it doesn't look right for the precious coral Coralliidae. So anyway, that's Paragorgia is my best guess.
Okay, great. All right.
There's also a nudibranch just to the right, a sea slug.
Oh yeah, can we see that sea slug there to the right, please? If we can get any more zoom on that. Great. It's certainly kind of translucent like the one we saw earlier. Okay, thanks. This is our max zoom here, so. All right, I think science is good. All right, so we're going to do a little pilot rotation here.
Can we get a zoom on these urchins?
Yeah, let's let pilot get settled here, and we will zoom in. Oh, I see one to the lower right there. Is that what you're talking about?
Yeah, I think maybe all three of these might be the same. We're just going to try to get it down to species or genus if we can.
Great. Yep, we can zoom in on those. Okay. So yeah, the ship's gonna have to make a move here. So we just got a few minutes, or a few seconds. Yeah, I think that should be good if we gotta go. Okay. I think these yellow things are going to be some more of the stony corals, but let's maybe just take a quick zoom there. Yeah. Oh. Yep.
So this is another one of the yellow corals that we've zoomed in on several times today. We think is Dendrophiliidae is the family. Could be Analopsamia, but it's potentially a little bit different. And I think there's a different color morph of this over to the right, the one that looks a little more orangey red.
Okay.
That one there. I think it's a very similar, potentially in the same genus. But I guess we'll
let's take a zoom
up to zoom in on it.
Yep. Yes, please.
Right, so this is another hard coral. It's sort of the deep water version of the stony or hard corals that are in shallow water that build all the reefs that people love to snorkel on. In deep water, a lot of these forms, or at least some of the forms, are fan-like, and these are non-photosynthetic hard corals. And this one here is in the family Dendrophiliidae, which probably doesn't mean anything to anybody but coral people. But this is a type of coral that we often see in a very low oxygen environment. To me, it almost seems to be a low oxygen specialist, because you can pretty much predict when you're diving at a certain depth that you're going to see some of these types of corals. So very, very interesting. My guess is it's some type of Analopsamia or hard coral.
Great, thanks, Chris. Yeah, and our oxygen is still pretty low here, 0.7 milligrams per liter. This has been the lowest oxygen levels we've seen on any dive here on this dive this morning. So. Yeah, we could look at this. I think it's urchin here by that other yellow coral. I think it's what it is. We can just take a quick zoom here. Maybe something that we've seen. I'm not quite sure though. Looks a little different than what we've seen.
Well, I'll take a stab at this.
You're on the hook here.
Since you're the geologist sort of stranded right now as Scott's finishing off his lunch. This one is a pencil urchin. It's in the family Sideridae, and it looks most similar to some members of the Stereosideris genus. It could also be in the genus Stylosideris or something quite different, but I think it most closely resembles Stereosideris. And I guess they get their name because their spines are very long and robust and look like pencils. I don't know if Bruce has any more information on that. He's a fish guy, but it's amazing how many interesting facts he knows about other animals as well. But somewhat similar to one we've got in Hawaii, not quite the same. So I'm going to go with Stereosideris, a pencil urchin.
Yeah, thanks, Chris. That's great. I don't think we've seen this yet, so that's great. Let's look at. I guess we'll look at these stars in the back next here as we're coming up.
And there's a little scorpionfish on the lower left just disappearing out of the screen. Scorpionfishes are not really identifiable beyond the family, so if there's time to take a look, that's okay, but
yeah,
if not, that's okay too.
Let's just do a quick zoom of the fish when we were swung over here. Bruce is saying it's another scorpionfish. We've seen 3 or 4 of these, I think, this morning already. Making use of some coral rubble there.
So this looks like the same species as the last one we saw. It's a little bit similar to a genus called Pontinus. It doesn't look exactly like that, so I don't really know if it is in that genus or not. The Pontinus are often larger than this. And who knows, maybe this is a species of scorpionfish that's sort of a specialist at oxygen minimum layer habitat, but we don't know.
Yeah, I see we're just getting word from the scientists in the chat room that the shallow water diving at Palmyra has been great today. There's a lot more biomass and fish than usual. So Jenny Castile is speculating perhaps the low oxygen levels down here are forcing lots of animals up to the surface. That would be interesting. We're still quite low here at about 0.7 milligrams per liter. I think that's a good. Science is good if we want to go back and look at those stars.
So one thing I just thought about as Bruce was talking about this fish, and I never really thought about it before, is, when we wind up going to higher altitudes where there's lower oxygen content in the air, such as in Denver, for example, the people there, the athletes there at least, they actually start producing more red blood cells to compensate for the lower oxygen. They need to have a little more efficient carrying capacity. But I'm just curious about these fish that are living in such a low oxygen environment. I'd be interested to know whether or not they have a larger percentage of red blood cells as well in order to deal with the very low oxygen levels they're living under.
Yeah, it'd be interesting to think about what kind of adaptations they may have to live in these levels. We're a little bit surprised how low these were this morning. So we got a nice view of this sea star here. I think science is good if we want to zoom back out. And we're hearing you some.
So this I think is Mediaster, and I think if I'm remembering Chris Maw's lessons properly, there's a change in the plate pattern that's right between the arms, and it was very obvious in that close-up, and I think that's very distinctive of the genus Mediaster. And it could be Mediaster ornatus, but I have to go back and look at my notes that I was taking furiously while he was telling me about these animals, but I think that supports that sea star was Mediaster ornatus.
Okay, great, thanks. This looks like the same thing to you, Chris?
Yeah, it does. And you see right in the crook of the arms, I think you can see a change in the size of the plates there. So they get smaller.
Yeah.
And it looks like there's almost a chevron pattern right in the crook there. And that's what he was pointing out to me. That's the type of feature that he is aware of. Most of us are not aware of it, that you actually need to pay attention to in order to identify these animals. So that's marvelous, and I'm sure that he can really come up with a good idea on this once he sees these images.
Great. All right, science is good. I think we've seen anemone before, so yeah, we can keep moving here, I think. Yeah, let's just take a quick zoom there. So we got a little time. Okay, they look like they're the same from here, so.
Could we get a little closer? Great.
Sorry, say that again?
So I'm going to take a stab at this again from memory. I think one of these was collected by some of the guys who are diving here. Maybe Bruce, you even collected this one back in 2005. But I think this is what Rich Mui might have identified as micropyga. It looks like an echinotheriid, but if you'll notice, the spines are much shorter, and there's quite a lot of them, and they look more stout. They don't bend like the echinotheriid spines do. And then micropyga typically has really long tube feet, I think, but I'm not really seeing that on this one. But I think this is what he might have called micropyga. I'll try to look back through my notes.
Yeah. Science is good. Yeah, we're certainly seeing a lot more rubble here, a lot more different sizes of sediment and rocks coming down from above where we are. Yeah, those are those tubes there? Yeah, let's zoom in real quick to see if they are. It might just be pieces of coral that have moved. Yeah, I think this is. Oh, I don't know what that is there. Yeah, what are we looking at here? Anyone?
Do you think these could be urchin spines?
Pretty big.
I think there's one there, Bruce, that looked like it was a tip of an urchin spine with the striations, but I don't know about the other one, the larger one. Maybe. So these plate-like structures, I'll take another guess because I'm definitely not bashful about guessing on stuff. This looks like these are the calcified plates from a type of algae that lives further upslope called Halimeda, or one of the relatives here, and it's one of the major sand-producing algae, at least in Hawaii here. I'm pretty certain that that's what a lot of these kind of smoother, very white discs are. So when the algae dies, then the tissue dissolves around these plates, and the plates fall free from the algae branches, and then it'll eventually be ground up into sand. So really interesting, good close-up.
Great, thanks, Chris. Yep. Science is good.
And if these larger elongate things are urchin spines, they're not from one of the urchin species that we're seeing here at this habitat. They must be something tumbling down from shallow water like the Halimeda fragments, too.
Okay, great. Thanks, Bruce. Yeah, we can take a look at this star here, I guess, just real quick, if we got a second. Okay. All right, that's good. I think it's the same star we were seeing. And then if you can you pan back to the right just a little bit? It looked like there was something on the cliff. There was a little bit different colors. Zoom out a little more. Yeah. Oh, that's maybe it's just another stony yellow coral. Okay, I think we're good.
Hey, Del, this is the web coordinator. Do we have time for a quick question?
Sure.
This is a question for the pilots.
All right.
From Michael R. Church on Facebook. He's wondering why it doesn't look like there's much current right now.
Well, that might actually be a better question for the scientist.
But can you confirm?
Yeah, there's not much current at the moment, no.
Yeah, so that's a great observation. So one of the things we've been seeing is that there's quite a bit of sediment here in the backside, and not a lot of bare rock exposed, and that makes sense given the kind of currents that we're seeing. We are kind of on the southwest side, and the reason we're parked here today is actually giving us a little bit of a lead from the weather be behind the atoll, and so we may have a similar effect with the currents here. That this is sort of the lee side of the current, so we're not seeing a lot of strong movement of the sediments, and the ROV is not experiencing strong currents today.
Thanks, Del. I have one more question as well.
Go for it.
And this one's from Twitter, and I can't really understand what the name is, but the person was wondering about what type of danger that the high pressure poses for the ROVs.
Yeah, so the pressure, the extreme pressures we see at the bottom of the ocean pretty much can cause implosion, which has at that depth, if it suddenly goes, we have a lot of housings, high pressure housings that can withstand the high pressures, but if one were to collapse, it would essentially be a bomb at the bottom of the ocean, which would be catastrophic, and it would destroy the vehicle. So all the housings, a lot are made of titanium, which can withstand the pressures down there. So about 6,000 meters, we see about 9,000 psi. So that's what a lot of our equipment's rated to.
Thank you, pilot.
Welcome.
Yeah, let's just go and take a quick look here. So it's another, it's like a black coral here. Yep. And so, yeah, if we could see the base, that would be great. We have one of our black coral experts here. I'd love to see the base of it if we can get an angle on it.
So Tina has ID'd it as a hexapathes.
Great, thanks, Gina. We moved a little bit, so we'll get another view of the base here, perhaps, if we can, for you, though. So, is this seeing suggestion of a collection? Is this something people are interested in, or?
This is Heteropathes black coral, by the looks of it. So some folks have been suggesting it'd be interesting to collect.
Suggestion it's a different species than the Mariana leg, it looks like, from Tina at least. So
Tina would know, she's the one.
Yeah, so here's a little better view of the base for you, Tina. Trying to get a consensus here.
Yeah, we have one request. I'm just trying to see if there are any other requests.
Can we get it, while we're here though, if we get a better zoom of the polyps maybe?
So I can't see that any black corals were collected on leg 1703. I'm not sure what the situation was on 1705.
That's us.
1702, I'm sorry, thank you.
Yes.
Yep, sorry, Dan, one more second. Dan, you think we're stable enough for a collection? Copy that. I think we could get the top section. Thank you. I didn't get a sense of how tall it was, and I'm not sure if the base would dislodge. Dan, can I talk to you? If you find that when you put the gripper on it that you can just shift it and it comes loose from the rock, go ahead and just grab it. Otherwise, just cut the top. If it's possible, that is a character they use, but I don't want us to have to wrestle it. It's just if it comes easily. Thank you. We haven't collected a rock, so this is 2. Yep. Yeah, you would have had to, yank it from the base. And I can see now from seeing the angle of the arm that that's not a good call. Yeah, these are really hard skeletons, polished black, so I think you should be able to cut it though. Excuse me. Yep. Oh, you got the base anyway. Great. I thought it might come loose from that carbonate if you moved it at the right angle. Nicely done, pilot. It seems that you saw a benthic ctenophore while I was at lunch.
It seems.
Well, I mean, that's what it says in the chat room. Dougal Lindsay wrote, wow, go get a copy and you guys see a Lyrotius.
But we didn't zoom in on it. That was my fault.
Oh, okay. Didn't realize
we were actually doing some questions. So
got it. So you saw those very large rocks when I left?
Yes.
Just looked like what, dislodged?
There were some dislodged, but
blocks.
then off to the right of where you were looking, there were some that seemed to be more in place, more of a little cliff. So.
Hey, Scott, I have a question from Twitter that you might be able to answer.
Okay. Amy, just stand by a second. Dougal, I see your comment, and I will absolutely video one if we see it. Go ahead, Amy.
So I had a question about what types of birds we're seeing.
Oh, yeah. So I was going to confess to Dell here that I may have been a little late coming back from lunch because I took a few minutes to go out on the deck. We are basically sitting right off of Palmyra Atoll, which is a great place for seabirds, and I am a birding fan myself, and so I wanted to have a look at the island. And it's got lots of trees. You can see the beautiful blue turquoise water over the coral. And there are at least 4 or 5 different species of birds flying around. So there are fairy terns or white terns. There are brown noddies. There are brown boobies, and I believe masked boobies, and sooty terns. So I've at least seen all 5 of those flying around the ship. And up on the bow, there looks like there's some juvenile boobies who have taken up residence on the mast up there, and a variety of others that are flying by trying to coax them to get off there so they can have a turn to sit down, but no doing.
Ah.
Thanks so much, Scott.
Okay. I'm having a question from Shore asking about dandelions, and I'm not sure what she's referring to. I see several of the larger kind of third urchins, and I see a much smaller plesiodiademid urchin, but I'm not seeing any of the dandelions. Typically when we refer to dandelions, we're talking about a kind of siphonophore, rhodalid siphonophore. And we've seen some siphonophores swimming through the water column, but these rhodalids kind of use tentacles to anchor themselves to the sea floor. And I did not see any, Tina. Yeah, I see the star lower center. That looks different. Unless it's really small and the arms are curled up, but we'll see. Oh, this one. The one on the lasers? Yeah, okay. We were looking at something different. That's okay. This looks like a brittle star. We've seen lots of these, actually. Yep. Yeah. Thanks. Looks like another one of the midwater fish there, the bristlemouths. We've seen lots of those, typically in the water column, and they may be descending and just kind of intercepting the slope of the island here. So I see 2 of these asteroid sea stars. One's much smaller. It's kind of at 5 o'clock from that first one, the one that we're about to zoom in on. That's by the lasers. Now I'm thinking this might be what you were just getting an education about when I was coming in. Chris was schooling you on how to identify these, wasn't he?
Yes, he was.
Great. So you're going to tell me what this is.
I'll tell you how to identify it, then Chris can repeat those funny-sounding words he said.
Okay. So this looks to me like it is indeed what we imaged, actually several times now, I think, on this dive.
Yeah, so this is the same thing we were looking at when, as Chris was describing earlier, the change in the size of the what do you call scales?
Yeah, he was calling them scales.
Yeah.
All right, that's good, pilot. We have seen this. Thanks.
Hey guys, this is Mary Wixton, and you were talking about those benthic siphonophores, and you'd think that most of those things would be poorly known and only from recent expeditions, but in fact, they were well illustrated by the famous biologist Ernest Haeckel, one of the foremost early exponents of Darwinism, in his book, let's see, what year is that? 1904, Art Forms in Nature.
That's interesting. Yeah, certainly some of these do have an artistic form.
The golden one and the red one right above it. Oh, you're going to want to go back to the right side after you're done, because I see a request that we had earlier. It's on the skeleton. That's the benthic ctenophore. Yep. So Dougal, we found your Lyrateus, so we'll get a look at that in just a moment. So the question is, so here's a siphonophore floating through the water column on our right. We're going to look at these two coral colonies and compare them to what we saw earlier. So the question is this big yellow gold one, is this a Acanthogorgia again, or is this now going to be a different kind of plexaurid? And then the red one, we have to determine if that's a bubblegum coral or if that's a coralliid precious coral. So that is our current mission. Now, the Acanthogorgia that we were looking at earlier had those yellow zoanthids overgrowing it. This one does not. All right, so we'll look at this. These are tricky. Again, I want to call this a bubblegum coral, but it looks so stiff that it might actually be a coralliid. And remember one we saw earlier, the tissue is stripped away and you could see these lines in the skeleton. And that's what's got me confused. So at least we've got a good image and we can send it around to our colleagues and see if we can determine that. That's good for the red one. Let's check out the orangey one. You can see on that red one there's some much smaller kind of like pimples almost. And that's probably siphonozoids, so modified polyps that aren't involved in feeding. They're much smaller. Was that a fish?
That's it.
Yeah, we can go for it. This is an Acanthogorgia, so we can go for that fish. Okay, we'll try to find that fish later, but right now we're going to look at this benthic ctenophore that's sitting on this dead colony. I'm not sure if it was a black coral or an octocoral growing from the side of the rock, but you can see there's something very dark sitting on the top of the branch. So we've seen many ctenophores in the water column. You can see they're reflecting comb rows. I was telling you earlier, it's the largest animal that can swim using cilia, locomotion by cilia. There's one group that's actually taken up a benthic existence. And so they've kind of abandoned the water column and their morphology has changed and they can sit, usually they're sitting on some other invertebrate. But there you can see extending to the left is one of its long tentacles, and you'll see little branches coming off there. So it's just like they're hanging out these long lines, have these very gelatinous appearance. And the mouth is actually facing down toward the branch that you're looking at. There you can see all the little side branches are called tentilla, I believe. So this is a benthic ctenophore. And we see on previous expeditions, I don't recall that we've seen it on this leg. We see some that are much, much smaller than this. I think it's a different genus, and they're associated with a lot of sponges and some corals. And we see, especially when we see the Walteria sponges, those glass sponges that have all the long spines coming out, they often have many, many of these really small benthic ctenophores on them.
Great. Well, I'm glad we got a good shot of it.
Yeah,
after
absolutely. Not right here. So that's some kind of octocoral. That wasn't a black coral colony, but you can see several of these bubblegum corals or coralliid corals. I think this skeleton we're seeing is the skeleton of that orange thing. I see one that's further to the right. That's a lot of the skeleton is exposed. Yeah, that might be a ctenophore.
Let's
Let's check it.
the ctenophore and see if there's a crab on that skeleton.
We'll do that in just a second, Mary. Just zoom in quickly and see. I think there's maybe a corallomorph inside that cave above left of the lasers. Yeah, so that's a corallomorpharian that's stuck in the back. Kind of looks like an anemone with white tips on his tentacles. And I can't tell, yeah, this gelatinous thing I think you saw there, Dan, in the foreground. I'm not sure what that is. There's 3 or 4 of them tucked deep into that little crevice, and then there's a couple more here just to the right of it in the crack. Yep, it's good for me, thanks. And Mary is pointing out there was a crab on that skeleton. I think the same skeleton that had the ctenophore on it. And I didn't notice it, Mary, so you'll have to give me some direction.
Just go out from the ctenophore.
Okay. You see it? Oh, yeah, now I see it. Yeah, great. There we go. There's 2 of them. There's 2 of them. Look like little spider crabs or...
And you're right, they are spider crabs.
Oh, good. Let's see. As you can see, a bunch of very small hydroids also growing on that branch. So you can see when the coral dies, the skeleton is taken over by a lot of other organisms. I think I'm seeing the head of a polychaete worm there on the left side of the screen sticking out of a little tube growing off of the coral. And there's a small snail up there. Nope, that's a hermit crab.
Hermit crab, yeah.
Yep. Another anemone. And there's our Lyrotius again, ctenophore. Great. Thank you, pilot. Oh, okay. We got a pretty good view snap of that fish that swam behind the colony. And for you on shore, it looks to me like a giant goldfish, a little bit more slender. Maybe... I don't know if that's snapper shaped, but it's getting close to that. So tell me if that's something that's really exciting. No, I think we can move on, pilot. Looks like a primnoid fan down there. You see the fish? Oh yeah, different. Oh, this looks like a lizard fish. Bruce is not there. Astrid, are you available? I think Bruce is unavailable for the next hour or so.
Actually, the fire drill that we had was very quick, so I just came back in time to see this gold-spotted or green-spotted duckbill fish. It's Cryonema chrysares in the family Percothididae. It's a species that we see often at these shallow dives. a gold-spotted duckbill fish, Cryonema chrysaris.
Bruce, do we see one of those at Jarvis as well? Do you remember?
Yeah, we've seen these on just about every dive that we've done in this 400-meter to 250-meter depth range. So, yes, we've seen them on recent dives in the shallow depths. They're pretty much ubiquitous throughout the Pacific, tropical and subtropical Pacific.
Bruce, I'm also not sure if you had returned already, but while we were looking at one of the coral fans, a fairly large fish swam behind us, and we only got a slight clipping of it on one of the side cameras, one of the, I think, manipulator cameras. And it kind of reminded me of a snapper, but kind of orange in color, or more like a goldfish color. Does that mean anything to you?
I didn't see the fish. I was outside until just at the moment that I called in. It sounds like you're describing Atoll snapper, which is the one species of the Atoll snappers, the deep water snappers that we've seen here in the Line Islands. Kind of a reddish gold color with a little bit more of a squared off tail than a lot of the snappers have. And it seems, I'm wondering if that's a species that's somewhat adapted toward existence in the low oxygen habitat. But that's pure speculation. I'll try to, I don't know if that would be on either one of the video streams, if I could scroll back and try to find that, or if it was seen on a different camera.
No, Bruce, it was seen on a different camera, so you'll have to wait until the replays tonight. All right, so we're looking at...
Okay, thank you.
Yep, sure. We're looking at what is probably a dead stony coral skeleton. You see that yellow coral down below. This is probably the same skeleton. And there's this urchin perched on it, and he's probably grazing over some of the material that is now overgrowing that skeleton. Thank you. Let's just nudge right and have a look at that brittle star that's coming into view. Okay, we can't see the disc, so that's fine. And there's that yellow stony coral that if you remove the soft tissue, you would remove the yellow color and you'd see just a white skeleton like above. Looks like there's a sponge, a spiky-headed sponge. If you just zoom in through that coral, it's going to be slightly to the left of the screen. Okay. Oh, I think it's an anemone. It's an anemone, and we're looking at it from behind. It's attached to a branch facing away. Thank you. That's good. Yep. It's a larger one. And it does look like it's a brachyurine crab of some kind. Mary, I don't know if you can see enough there to say anything.
Can't see him yet.
Yeah. You're not going to see much of him, Mary.
There's a time delay, so where, you mean to the left of that yellow coral?
Yep, it was in the crack. Yep.
Yeah, it's another member of the spider crab group, but that's about as far as I can tell you.
Yeah, we didn't get a great view of that one. Thank you.
Yeah, still pretty low oxygen here, 0.73 milligrams per liter. I was just looking at the EK60 sonar. It looks like there's not a lot in the water column until about 200 meters, so we may be in this low oxygen zone all the way up in our dive today, or maybe just barely get out of it at the top here if we're lucky.
So one of my colleagues asked a question when we were outside for the fire drill. Are we on the east side or the west side of the island? Since there was no dive plan, but I'm not sure which side we're on.
Yeah, sorry, Bruce. We're sort of on the southwest slope.
Okay, he commented that the equatorial undercurrent, which runs from west to east, is impinging in this area, and that may have, and then being swept up by the slope of the island, and so that may have something to do with the depth of the oxygen minimum layer that we're seeing here.
All right, thanks, Bruce. That was it.
Another one of those urchins that we just saw on the coral. Another anemone on the base. Oh, wow, look at that. There's at least 2 of these Acanthogorgid corals tucked up underneath that rock. Huh. It's interesting, to think, how did these corals get there in the first place? And so the first thing you have to remember is that there is a larval stage. So it's a small ball of cells, has cilia, so it can push itself through the water column. And it's going to look for a place to settle. So something that is clear of sediments, has an appropriate substrate, and then will start to grow. And so I imagine that there's enough current whistling through here. Yeah, so that looks like an Acanthogorgia. Again, you can see the relatively thin polyps extending from the relatively thin branch. And I don't see any crabs or anything on it. There's a couple of things that could be hermit crabs. I see 3 white dots, and based on some of the other close-ups, that's probably what that is. Thanks, pilot. That's good. On the left side of this rock, there's something orange. It might be a sea star draped over something. I think you have to come out just a little bit further. There he is. Just came into view, and he looks like he's in a feeding posture, and you're in a good position. So. Well, if it is feeding, it's doing a good job of hiding what it's feeding on. I can see to its right there's an open tube, which probably had a worm in it, so there could be something like that underneath there. We see there's a number of other corals growing on here, so it could be feeding on one of the corals. And again, we're seeing these stubby little white finger-like projections, and we believe those to be really small hydrozoan corals, or hydrocorals, which is not something we've been seeing very much of, any of, on the cruise, except for those stubby little white things. But if you dive in the Caribbean and you've seen fire coral, that's another version of a hydrocoral. Yeah, so what exactly is going on there? I do see there looks like a tentacle going up above it through its top middle arms. It's disappearing into a tube, so maybe it has a hold of whatever that is. Indeed.
Huh.
I wonder, that could be, a tentacle of an anemone, for example, and maybe it's feeding. Yeah, I think it's one of those corallomorphs. You see the white knob at the tip?
I see it right
There
now.
we go. So I believe that sea star is feeding on one of these corallomorph, which is a relative of the sea anemones. It's actually more closely related to the stony corals, and some people in the past have called them naked corals, and there's been some research done to find out, are they truly, quotes, naked corals? That is, do they share a common ancestor? Did one evolve from another? And so on. Great, good observation. Move on. I know Chris will appreciate that. Yep, let's do it. So among the scientists that are in the chat room is Jen Cassell. She's from University of California, Santa Barbara, and she's part of the Palmyra Atoll Research Consortium that includes UC Santa Barbara, Scripps Institution of Oceanography, which is part of the UC San Diego, the University of Hawaii, Bigelow Labs, USGS, and Stanford. And she said that they're thrilled to have this deep water work and exploration that are done here because they can't access these deep habitats with their normal operations and tools that they have. So thanks, Judd. There's another Hexapathes, the black coral fan at the bottom. We collected one of those earlier. We've seen several somewhat deeper on the dive. Still lots of these urchins moving around. We saw one moving pretty quickly earlier. They can move if they want to.
Hi, guys. I'm going to have to sign off, but just a note. The only squat lobsters that are known to be tolerant of low oxygen belong to the family Munity. You don't get too many records of Chirostylids or occasionally Immunodopsis in low oxygen. So if you see a squat lobster that has bug black eyes, it's probably going to be in the Munity. I'll see your pictures later. Sorry, I got to sign off. Bye.
Thank you, Mary. So that's kind of interesting, and that may explain why we're not seeing squat lobsters on the corals today.
Yeah, I think we saw one in a rock.
And
earlier. Yeah.
Mary came in, and I think she told us we had identified it incorrectly, that it was a squat lobster, but not a chirostylid squat lobster. So she's saying they're more the munid
I see. Yep.
family. Stuff for Jadell, I think. It's all rocks and sediment.
All rocks coming downhill here. So. Yeah, so you know these islands really are a source of sediment, not just for the slope, but in this area, much of this reef material or carbonate material eventually makes it out even in the deep water and the basins here between Kingman and Palmyra.
Oh, sorry. Can I talk, or are you talking to... Yeah, no, I'm sorry. I saw you were communicating up there. Can you back up? Is it too late to back up a little? I don't see it yet. It was kind of dark gray, black, blue, and it's hard to see, and it was on the top of the rock, and I thought it was a shadow. But if you just keep going, there, it just came into view, the lower left of the screen. And I thought it was a shadow on the rock, but as we were passing over, I saw that it actually was growing up off the rock. Understood. It's almost exactly to the right of the lasers, but just going off the screen. You see there's kind of a blackish structure on the top of that sediment dusted rock. Yep. Just above the lasers now. Copy. One hour left on bottom. Copy. Yeah, let's make our way upslope. Thank you. So this is a black coral. This, I think, is a different kind of black coral than we've seen today, so nice to get that observation. That's what I thought was going on. It was something different. I'd rather get upslope. Thank you. We're at 385 meters depth, climbing up the slope of Palmyra Atoll. We're on the south side, the southwest side of the atoll. And let's see, our temperature now is almost 9 degrees Celsius at the bottom of the ocean here on the slope. What's that, 1848 degrees? Light sweater weather. And oxygen still below 1. It's at 0.8 milligrams per liter. See one of the Pom-Pom Anemones now, just the laser's just crossing past it. Still many of these sea urchins and sea stars. Sounds good, Dan. Lots of sea stars down there. And presumably they're feeding on something in the sediments.
Mm-hmm.
It seems that most of the sea stars and the urchins and the brittle stars we've seen today have been patrolling the sediments. We haven't seen all that many on corals, although we haven't seen all that many corals.
Right. No, I think that's fair.
And pilot, if you in the front row are comfortable, if you want to introduce yourselves.
Yeah, so we're all from RV team here. Sean Kennison is sitting pilot right now. We're all from the Global Foundation for Ocean Exploration, or GFOE. And I'll let everyone else introduce themselves.
Over on Navigator currently is Andy Lister, and as Sean said, with GFOE. And this is
Thank you. That's indeed what it is. So another dead skeleton of, I'm not sure what kind of coral. This actually, the internal parts of that skeleton suggest bubblegum coral to me, but it may be that's just so overgrown. That's good. Thank you.
Yeah, we see a number of pieces. This kind of thick skeleton we were at lunch too that was dead. So seems to be quite a bit of it.
So we were looking at the those pink colonies earlier, and I'm trying to determine whether it's a bubblegum coral or if it's a coralliid, precious coral. And for some of those species, they can be a little bit hard to tell apart, which is a little bit surprising given they're in different families. But both of them, many species would have white skeletons underneath it. But so too do the Sclerotinian corals, those yellow ones that we've been seeing. But the bubblegum corals, the structure of the skeleton, I won't call it spongy, but it's got this interesting matrix when you sort of break it and look at it. And that's what that reminded me of.
Almost looked like bone there, okay?
Yeah. But it's not as rigid. And the flip side of that is that looked pretty rigid.
There was a different kind of fish in the cavern. We've already gone past it, though. It could have been one of the bariciform fishes, maybe a barrax, or it could have even been a roughy.
Sorry, Bruce, I did not see it, and I will keep a closer eye. We're moving a little bit faster as we try to get to a shallower depth, but if we see it early enough, we'll definitely get a good view of it. Looks like we're passing over another Acanthogorgia that's largely overgrown with zoanthids. White primnoid colony on the left side. More urchins, more primnoids. One of the things about corals growing on the carbonate, and Del, maybe you can correct me on this, but my impression is that this weathers much more quickly than the basalts and the manganese encrusted stuff.
Yeah, you can see that this is breaking apart. These blocks are rolling down this steep slope. Definitely maybe not as permanent or as long-term a home as we saw in some of those thick encrusted volcanic rocks. We've seen at deeper depths.
So that's definitely going to have an impact on the sessile fauna that are growing on it if the rock underneath them is kind of eroding away, becoming weaker.
Right.
It's not a great home. So that's one of the factors likely that is affecting why there aren't more coral right here. The other is just the amount of sediment, maybe that we're in a bit of a shadow of the main flow of current. Can't tell if that's a fish above the laser, or is there, I think it's just rock.
Yes, of course, we're here in the Line Islands, and we've got these carbonate reefs and atolls that we've been visiting here, Palmyra today, Kingman tomorrow. And these are really built up on a fairly high-standing bit of topography compared to much of the abyss around us. And of course, the original origin of this high topography is all volcanic, but in fact, we've built up kind of a large section of crust here. And folks have looked at the gravity in this area. We can see that this crust is actually thick enough that we think that in some ways this high-standing topography is being isostatically compensated. That is kind of floating like an iceberg. So if we've got low-density material that's thick enough, these islands will sort of float up here. And that's important because if we've got high-standing topography for long enough at these shallow depths, we can accumulate more carbonate. Whereas in the deeper waters, if we get deep enough, carbonate rocks start to dissolve. And so this is really why we've got this big sequence of carbonate reef platforms here.
Just passed over another one of the black corals, like the one that we collected. A fan. It's kind of dark in color and has some branches that stick out in front of it. It's amazing to me to think about how many coral skeletons it took to grind up to make all this sediment.
Yeah, it's a long time. So.
Yep. It's another Acanthogorgia colony sticking off the rock on the right. Not only that, but I guess it's also interesting to think about when there's a lot of discussion about carbon dioxide in the atmosphere, and these animals are sequestering that carbon dioxide by taking it out of the water and incorporating it into the skeleton. So then it's stored in the skeleton or it's stored in these sediments. And so you may often hear about various animals in the ocean or the ocean itself being an absorber of carbon dioxide, a place that CO2 is sequestered. Another Pom-Pom Anemone. Maybe the word I was looking for was carbon dioxide sink.
Sink,
yeah.
Yes.
This is a fish that is swimming along the sand just to the lower right of the lasers. Oh, two of them. There's one a lot closer to you as well. They're right in the foreground. I don't know which one's easier to get.
So I think these are little dories in the genus Cytomymus, different genus and family than the Oreodories that we saw before. Yes, they definitely are. And I don't know of a better common name for
these. Went over a flatfish, Bruce.
A small dory species.
Stop on flatfish, please. I don't know if it's a tonguefish or something, but there's a little flatfish sitting there. He's at the top of the screen.
Good eye.
I don't think this is a tonguefish, but I could be wrong. The tonguefish don't have a distinct tail fin, and this one looks like it does. It also has that fin it's actually on the side, even though it looks like it's sticking out of the back, the pectoral fin. That posture, yeah, it's definitely not a tonguefish because I can see it's got a squared off tail, and the dorsal and anal fin slightly disconnected from the tail. This is a right-eyed flounder. You can look for the gill openings, and you can see those moving. It's breathing rather heavily in this low oxygen environment. I think this is in the family Samaridae, and in the genus Samariscus. Don't know if it's a species that's found in Hawaii. There are a number of these species in the genus Samariscus that are known from New Caledonia and points to the west, and they're also found down in the French Polynesia area. We're probably looking at a new record of this family for the Line Islands, and it seems like this is also a family that continues to have new species described. I'm reevaluating after saying all of that. It still doesn't look like a tonguefish, but on the other hand, if there are other people watching Astrid or anybody else that has a different opinion, I'd be open to that. I do think it's probably in the family Samaridae.
Bruce, you're starting to sound like me, equivocating between the bubblegum coral and the corallid. All right, that's a great video. Thank you. I noticed another Galatheid crab stuck underneath that little edge. Yeah, go ahead, Bruce.
I was going to say it's just a little bit of a problem of trying to make these identifications from the excellent video that the Okeanos gets, but without having the specimens. When we see so many things that could be new, or at least new records for the region, we do have to speculate a lot and qualify what we say, because we can't really know what a lot of these things are unless we have the specimens. I do envy you coral biologists, because you can snip off pieces and take them back to verify what you're seeing.
Well, that's well said, Bruce, and we'll remember that envy when we're whining that we don't have more. You're right, at least we have some. Is that a fish, or it looks like a snake? It's on the right side, right quadrant. Yeah. I wonder if that's one of those snake eels that I kept calling snakefish the other day. Yeah, I think so.
Yeah.
So, yeah, I don't have much to add to that. You got it. It's a snake eel. They're difficult to identify. If we can see fins on the side behind the head, it's probably an Ophictus species. If it doesn't have the fins on the side behind the head, then it would be in another genus. Although,
yeah, it's hard to see.
After looking at this, I'm reevaluating what I just said. It's got an odd color pattern. Up toward the head, there's a sort of a saddle of dark pigment that comes down behind the head, and then there's a light area behind that saddle. And this color pattern looks like something we identified previously on a previous dive as the family Clopsidae, which is the false moray eels. Yeah, now we can clearly see it after we zoomed in. So after getting a closer look, I think this is in the family Clopsidae, in the genus Clopsis. And so everything that I just said, with all of my equivocating, I take back about it being a snake.
Sure moves like a snake.
No pectoral fins.
Okay.
So, Bruce, does this look like the other clopsids that we've seen? This one looks different to me. I'm not sure I've ever seen this one before, or any pictures of it.
We saw one with this color pattern on a dive a few days back. It was kind of among some corals or other things, so we couldn't see it as clearly. But the color pattern on the head, we definitely saw one of these on an earlier dive.
Looks like the crab's cleaning off its eyeballs. Eye stalks, more appropriate term. It's a bunch of seedy or bristles lining those claws. You can kind of use it like a brush to clean off. Well, that's good. A couple of interesting fish there. Sure looks like a snake.
Yeah, it's hard not to call it a snake.
Yep. Is that another flatfish, or is that a rock? That must be a rock that's protruding out of the sediment, huh? Even though it sure looks like a fish, right in the middle of this chute. It's a rock. Oh, we got to see one of the brittle stars moving down there in the lower part of the screen. No, that's okay. Let's head up slope. Thank you. On the lower left of the screen, you see there's a stick coming off the rock. We'll see what's it. Oh yeah, so that's going to be another black coral. First time we've seen this. You see it's got a coil to it. That'll be the genus Stichopathes. We'll just get a quick look and verify that. But there aren't too many things that it could be with that shape and size. We probably should just be able to see almost like pairs of tentacles coming off the axis. Yeah, there you go. So that is indeed another black coral, so-called because the skeleton is black. Thank you. Whip-like form, quite common. There's a lot of taxonomic work that needs to be done on the whip-like black corals. They are currently all classified in the genus Stichopathes. That's not entirely true. There is another genus Cirropathes, but they have a different polyp structure, so we would be able to separate them. But the ones that are all called Stichopathes, based on genetic work, are clearly not all related to one another. So there's some taxonomy work that's going to have to go on, some new names, some new groups applied to better classify that diversity. And that's a colony of Narella, a primnoid octocoral. We've seen several of those today. In fact, we haven't seen too much diversity in the octocorals. I can think of the Narella, Anacanthogorgia, and this pink colony that's either a corallid or a paragorgiid. I'm not sure that we've seen any others. No sepens today. Les Watling from the University of Hawaii is noting that the dissolved oxygen is now above 1, so at least it's tolerable by most species, but very interesting how low it got, and it'll be interesting to see in the final 35 minutes or so of our dive if the oxygen goes up and if that changes some of the organisms that we're seeing. Note a few other people who are in the chat room who we may not have mentioned so far today. So Asako Matsumoto from Japan, Emilio Petruncio is from the U.S. Naval Academy. Sonia Rowley is from the Bishop Museum at the University of Hawaii. Thank you all for joining us and helping us with the observations today. There's another one of the green eyes, I believe that was. Let's get a quick zoom on it, pilot. And these may be polychaete. Let's look at also these brown tufts. We can look at the fish first, but I want to get a look at those brown tufts on the rock, because we saw a lot of that stuff on the slope of Jarvis Island. It turned out they were polychaete tubes, and it'd be interesting to know if they're also here.
Well, this is another one of the gold-spotted duckbill fish that we saw before.
Fish,
a very common type of fish down here at these depths. It's a spiny-rayed fish. It's got 2 fins on the back separated by a little gap. The first one up toward the head has spines between the membranes, and the second one has soft rays. When we do the deeper dives on the Okeanos Explorer below 1,000 meters, we don't see many of these spiny rayed fish. The spiny rayed fish are more characteristic of shallow water habitats like shallow coral reef habitats. And it's
That's what I'm looking for.
A gold-spotted duckbill fish.
Thank you, Bruce. Do we have any more zoom? So we're looking at these tubes, yeah. So if you see the ends of the tubes, you see those two antenna-like structures sticking out. And I believe that these are not antennae, but tentacles of worms or cirri of worms. So this whole cluster of tubes that you're seeing has been built by polychaete tube worms. And they're all kind of glued together to each other, and then coming off of them are hydroids. You can see there are brittle stars crawling on them. I see an anemone growing there. I think I see a hermit crab in the background. And the last time we saw this on Jarvis Island, I've not seen them right now, but we saw a number of the caprellid amphipods or skeleton shrimp, also called noddies. So basically, each one of these tufts seems to be its own little community that's attracting all this other stuff to grow on it. Thank you, Pilot. And as we pull away, you're going to see there's a number of these different little clusters of polychaete tubes. And on the Jarvis Island slope, we were seeing literally thousands and thousands of these all over the seafloor. So it was really interesting how it was creating the structure for additional small organisms to live in and among. You can see there's several of them there going out on that flattish slope there. That's tilted down towards the right. So that's kind of interesting. So now we've got the polychaete tufts. We saw that on Jarvis Slope, and we had that area where we had all those Spatangoid urchins really clustering together on a ledge, and that was another thing we saw on the Jarvis Island slope. So some similarities at the common depths between these two areas. Both in monuments, but separate units.
Mm-hmm.
Not sure what that black thing is there on the right side. Is that just a rock or a piece of wood?
Oh, yeah.
And is that a fish on the top of the rock? Is that what you're pointing to, Annie? Okay. Yeah, this could be a rock, and excuse me. If it is a piece of wood, there's various organisms that are associated just with sunken wood. Oh, it actually looks like it's a bit of a palm frond or something.
Yeah, it does.
Right. Yeah, with some urchins on it. Okay, Pilot, there's a fish on the top of the rock. It may have been another scorpion fish. Do you slip away? Oh, well, there's a different fish, but spikefish. Yes, I was going to call it triggerfish. Yeah, let's have a quick zoom.
Yeah, it's another spikefish, you're right, and they are related to the triggerfish in shallow water. These fish can change color, and this one might. When they're frightened, they get pale. Sometimes they're bright red like this with the white spots, and sometimes they're more pale. So calling it a triggerfish is not a bad guess because they are related and they're similar. We often see these around coral fans, but not always. So there seems to be a little bit of an association there, but it's certainly not obligatory for this. So the fish is kind of stunned, I think, by the bright lights of the ROV, probably wishing it would go away, a spikefish.
We're good on the fish, thank you. continuing to come up this slope. We're at a depth of 335 or 36 meters. Right now, we're trying to at least get up to the 300-meter contour. See if we can start to get into the bottom fish depths. And depending on how long you've been following us on the dive today, we have come to numerous places where we have seen some fairly large coral colonies, both octocoral colonies and stony coral colonies, and a few black coral colonies. But for the most part, it's been pretty heavy sediment drape.
Mm-hmm.
And it's more common to see the sea urchins and the sea stars, like we're just passing over now, brittle stars, several of these Pom-Pom Anemones, which seem to be just fine living on sediments.
As Chris Keller was mentioning earlier, lots of these little chips we've been seeing are these calcified algal chips that are coming down from up above. That's a lot of the larger sediment maybe that we've been seeing in some of these areas. And of course, we occasionally see much bigger blocks coming down the slope.
I think this is a pilot. Are you still listening to me, or are you busy? Okay. Just to the left, upper left of the lasers on the surface of the rock, white circle, excuse me, yellow circle with red. I believe this is a tremaster. We don't have to spend long on it. We've seen it several times in an earlier dive just to document it. Yeah, so I believe this might be tremaster. I think it was Mirabilis was the species name. So a type of sea star. And there was a bit of a surprise. Chris Ma had noted that there was a paper that came out last year that showed that this was feeding on some coral in the Atlantic. So we know that it can at least be a predator of coral. Thank you, pilot. Is that a large white sponge? It's just on the upper right of the screen. If so, it seems remarkably out of place compared to everything that we've seen today.
Yeah, it really does.
But we did just get over 1 milligram per liter of oxygen, if that means anything. Huh. Chris Kelly, would you look at that?
Well, surely Pomponi would be better at this.
Yeah, Shirley is not available.
If it's a sponge, which it may be, I think it would be a demosponge, not a hexactinellid. Shirley, are you still there?
I don't think she is. She sent me a message earlier that said she was going to have to leave.
Yeah, I'm not convinced it's a sponge even yet. I think it is, but it could also be some incredibly white piece of carbonate that had tumbled down and wedged in that little nook.
Huh. Okay. I think we're seeing one of those stylastrans. I've just been communicating with Asako Matsumoto on shore, and she's been asking about these little finger-like projections from the bottom. They're very white. And Chris Kelly, who you just heard there, had noted that they had collected something like this on an earlier expedition, and they believe it's a stylastran hydrocoral. And the one that's there above whatever this dish-shaped thing is one of the largest that I've seen so far. It's at an awkward angle there to us. Can't quite tell. Maybe if we look at the edge of it, Sean. Oh, sorry, I meant the zoom on the edge. I don't want to put you in a position where you fall behind. Okay, go for it. Go for it. Well, that's ideal. Okay. Thank you, video. We're getting some ID on shore. I'm just not sure what the acronym stands for, so I'll get that clarified in just a second. No, that's good. Thanks, pilot. Oh, wow.
Huh.
So Jen Cassell is suggesting that that is dead crustose coralline algae. Really, that large white thing. Get some clarification on that. There must be a colony there because I see a brittle star hanging in space. I guess he's hanging onto those worm tubes. Well, the debate rages and continues in the chat room on exactly what it was we were looking at.
So it makes it interesting.
Yep.
It's coming up on 320 meters here. A little little urchin party.
I wonder if they're feeding on something.
This is a
Yes,
kind
please.
of a nice little display where you can't necessarily go by color on these things as being a great indicator of species. I think these are all the same species, and you see a variety of color morphs here.
So I think there might be some seaweed, some kelp underneath there. I see something that's green and yellow. So perhaps this is some, or maybe it's some leaves from a tree or something that have settled down the slope, and these urchins are now consuming it. Many of these urchins, at least in shallow water, are herbivores in the sense that they feed on various kinds of seaweed and algae.
Okay.
Nice view. Thank you.
So this is the first one of these that we've seen, and it's the kind of thing I would have expected to have seen more of on this dive based on the submersible surveys. This is a grappo in the family of the splendid perches. It's in the genus Grammatinotus. Some of us just call them Grammies too. It's got a tail fin morphology of a species that we call Grammatinotus laisanus. We see some of these without the pale spot on the side and some with the pale spot on the side, and we don't know what that means, if they're different species or if it indicates males versus females or exactly what's going on there. These could be quite common at these depths. They're a small type of fish. Some of the larger snappers and other predators will eat these. it's been surprising that we haven't seen these before. And there's an absolutely beautiful squat lobster just below it. I'll let somebody else comment on that.
As soon as I saw the fish going over, you were still describing, and I was pointing at that beautiful lobster. Let's check out that squat lobster pilot that we just passed, just a little higher up. Kind of interesting, maybe a cup coral just down there. Bunch of polychaete tube worms, fan worms. Yeah, really pretty coloration. So I think this is what Mary was suggesting might be the munid squat lobsters. I didn't realize they came in such flashy patterns. We call it a squat lobster because it's kind of somewhere between a crab and a lobster. It has a reduced abdomen, that tail flap, but not so reduced like a crab that it's totally folded up and oppressed underneath its thorax. All right, that's good, pilot. Thank you. Dougal Lindsay is suggesting perhaps that's in the genus Baba munida, Baba munida. And it has a species name, Javirii. Jen Cassell followed up on our observation there of all those urchins feeding on the algae. That is interesting because they don't see very many urchins up in the shallow water here at Palmyra. So it's a little bit of a surprise to her to see so many down deep. There's a fish. I'm not sure if that's a midwater fish or something different. This dark one.
Well, this is another kind of fish that I would have expected to see in very high abundance in this area based on the submersible dive. It's a deep water cardinal fish in the genus Epigonus, family Epigonidae. They're similar to the shallow water cardinal fish, but they're actually not related. They were placed in the same family as the shallow water cardinal fish for quite a long time until they were separated out. And there's another little grappo on the bottom as well. So Epigonus, there's another one, two of them. They usually occur in sort of aggregations on high relief areas where the plankton, there's a good accumulation of plankton.
No, we're fine like this, and in fact, I think we can move. Sorry, Bruce, if I was speaking over you there. Speaking over you. I hit the wrong button. Now this has got to be a Leopathes. This is something we're going to want to see, especially because it looks like there could be a giant nudibranch in there or a pile of eggs or something. So earlier in the dive, I was telling you all about Leopathes black coral and how they're interesting, and one has been aged at, get this, 4,500 years old.
It's old.
Yeah. And then we zoomed in and discovered it was an octocoral. But this, no mistaking it, this is now a black coral. So that's a cluster of barnacles. How interesting. These are lepatomorph barnacles or like gooseneck barnacles. And what's in the center? There's something, I guess, oh, that's a crab arms, another very colorful one. They make their crabs very colorful at this depth. But Leopathes, there's a family, the Leopathidae. Currently, there's only one genus, Leopathes. There's 5 or 6 species that have been described around the world. And this is one of the popular ones for black coral jewelry because you can see the base of that skeleton is really thick. It also, and I was sort of describing this before, it has these kind of very gracefully curving branches instead of straight and angular. So I think you can get a lot of interesting shapes and pieces for coral jewelry. It's also a little bit different from most of the black corals because the thick parts of the skeleton are very, very smooth. And it's only the smaller branches that you can really see the spines. But otherwise, it's comprised of all these polyps, 6 feeding tentacles each. And you can see these colonies can get extremely large. And that's just a beautiful view of that crab in the center of all these barnacles. Copy that, Nav, and no pilot. I think that's good. Beautiful colony. Chris Kelly had said on one of our earlier dives, we thought perhaps we saw a white morph of Leopathes. This orange one is far more common. The white one would be something that could be of interest for collection, but we don't need that one. I love that little
Yeah, we
bouquet
might
of
still
barnacles.
see the white morph. I think they're here off of Palmyra. One of the interesting facts about Leopathes, Scott, just to add to the comments you made, are that it's one of the oldest or long-lived corals right now that's been dated by Rob Dunbar and Tom Gildeson and Brendan Rourke's group. And I believe that genus Leopathes in Hawaii, which is now Anosa, has been dated, a single colony was dated to more than 3,000 years old. It's still alive, so it's really incredible.
Thank you, Chris. Certainly seem to be scaling the mountain here.
Mm-hmm.
See more of these tufts or clumps of polychaete tubes. That's one that's just left of the laser. And it looks like several more coming up here. And I think this is about as shallow as we have been on this expedition, except perhaps for the very first dive in the Anu unit. I think we may have gotten up to 250 or so meters there.
Yeah, it's very, very similar here.
Yeah. If we look over to the left, there's a branch sticking out from the rock, but behind it there's something light blue purple. Okay, I will be calm and wait until you're ready to go there. Not sure if it's a sponge or a fish, or maybe it's another Lyroteus. I'll just keep guessing for the next 5 minutes.
Eventually, one of them will be right.
Oh,
wow.
Chris Kelly said we might see these purple octocorals. This is of some interest, and there may be our first cup coral. You can definitely see now that's got a cup skeleton on the base. Yeah. Oh, this is a really interesting blue color. Wow. This is not Victorgorgia, I don't think.
Whoa, look at that.
Yeah, it looks like two tussling crinoids. That's something you don't see very often.
Crinoid battle,
yeah. What's going on there? Chris, you have any comments about this colony? It's more blue than purple. Another one of the...
it's absolutely gorgeous. That's the first comment. And the second one is it's attached all along this branch here. It's got quite a large attachment for the size of corals that it is. You know, I have no clue. I guess it's a plexaurid of some kind, but that's the best I can do.
Yeah, no, I don't think it's a plexaurid. I think what's happened here is you've had a plexaurid die, and then this is taken over. So in fact, this is consistent with the way Victorgorgia grows. It's a Sclerotinian related to the bubblegum corals, and they can put out these really wide holdfasts, almost like a dripping wax, you know, and it covers the bottom, and then it can grow erect from that. And then again, you can see that cup coral on the right side. You can see from the side view it has this stony calcium carbonate pedestal and a single polyp on top. So we call that a solitary coral because it doesn't asexually bud to produce new polyps that all remain attached together. Okay. I noticed there were another one of those very large rounded plates on the left. We're not in a position to be able to look at it, but so although I said at first that was not Victorgorgia, I'm now thinking that it is, or it's something related to it. So it was an octocoral, if that wasn't clear. So where we're climbing right now, the steepness has changed quite dramatically. So the ROVs are making sure that the pilots are making sure we can see what's ahead of us so we don't go crashing into something. Still,
Could we zoom in on the sea star? This is a different one. I think it's Nepantia, the one that's going off to the left right now.
Stand by, Chris.
I don't know if you've seen one of these before. I had to step out briefly during the dive, but this is different than the majority that we've been seeing all day.
Okay. Seems like it has a little bit more of a membranous feel between the arms.
Yeah, exactly. I'm going to take a look at the pictures I've got. There's forgotten what the family is, but we were talking about this, Chris Ma and I were talking about this about a month ago in his office.
It looks to me...
there's two genera. There's Paranepanthia and Nepanthia, and I think this is Nepanthia. They both have these kind of larger skirts, is the way I kind of think of them, between the arms.
Right. Is it a mixed aster? Mixed aster day? I mean, I noticed it's got what looks like a little chimney or osculum on the top. That's good, pilot.
It's in its own family, I think. My memory is escaping me right here exactly what family it is. I'm going to try to Google it right now and see if I can figure it out.
If we're in a good position, we can look at that upper fan on the lower side of the screen, lower left of the screen. Yeah, there's two of them. It's the one above the white one I'm interested in. I'm thinking, based on the darkness of the skeleton, that this is probably a Acanthogorgia again. Just want to get a good look at that. And it certainly is. Again, I'm using the relative skinniness and tallness of those polyps. This one is a much lighter in color than the one we were seeing earlier in the dive. And still can't get a good look at this kind of crown of thorns, as I would describe it, sclerites towards the top, and none of the polyps are retracted, so they're not helping in that sense. You can see an anemone on it and a crinoid, and that's good, pilot. We can move on.
So Scott, the family for that sea star is Asterinidae, so it's not it's not a mix. It's not the other one.
Yeah, so yeah, Chris, that's not a family name I hear a lot about on our dives. I mean, is that a fish? No, that's a big snail, I think, in the center of the screen, above the lasers now.
It's a fish. Go with your gut, Scott. You nailed it the first time.
Oh, yeah, it is. Look at that. It's a little bat fish, huh?
Wow.
I recognize. It's got the little mouth. No, maybe it's a goosefish. Stand by.
Well, you're close. You've got the right order. It's in a different family, and we see these fairly often, again, at this depth. This is a gaper or a sea toad, or a coffin fish in the genus Chaunax family, Chaunacidae. This one's got a little different color than most that we see, so it'll be interesting to send the photos of this off to the experts. The color on this one makes it a little difficult to see, but there's a short lure between the eyes, kind of flattened down on the snout. It doesn't have the lure on a long stalk like the goosefish or the shallow water frogfish have. And the batfish have the lure actually underneath the snout, pointing downwards. You can also see that this has some large lateral line pores and pores on the head for sensing pressure waves and vibrations. It's an ambush predator, sits and waits for little things like that shrimp to swim by, like the one that just did, and then will engulf them. So it's a gaper, or a sea toad, or a coffin fish. A variety of common names are applied to the species of Chaunax.
Great. Thank you, Bruce. And Sean, as we're passing by, just have a look on the left side. I think it was a bunch of cup corals, all dead, but they're sticking off the wall. We'll just... Oh, they're alive. So there's one that's alive. Oh, I see. There's 3 more that are dead. But you can see there's the cups. They're made of calcium carbonate. They're secreted by the soft tissue, the polyp, and you can see in the foreground, just below left of the lasers, there's one that's got all the tentacles extended, so that one's alive. lots and lots of species. In fact, half of the known species of Sclerotinians, I believe, are these solitary forms. Great. Yep, I am. Thank you. So we're basically at the last couple of minutes of our dive. Maybe we'll get lucky and see a couple more fish here, or at least the benthic portion of our dive. You can stay tuned with us as we rise through the water column. And there's no transects today, but you still might see something interesting in the water column, although it's a short trip to the surface. We're at 304 meters depth.
Yeah, it looks like we're still a little bit below that layer in the water column where the sonars are showing lots of reflections. So I think our oxygen is up at near a high here, a little over 1 milligrams per liter again, which would be the lowest we've seen on any other dive so far. So.
Just want to make a little correction to the sea star and researching a little bit. It's the Paranapanthia. I guessed wrong. I had a 50-50 chance there from what I can tell. So it's not Nepanthia, it's Paranapanthia, that sea star with the skirt.
So noted.
Is that yellow underneath that overhang something different? That might be different from anything we've seen earlier. It just has a different feel to me. I'm thinking this is a stony coral. No.
Oh, bizarre looking.
Yeah. Is this a, what is it, Uchigemia? And there's a fish down there. Is that a snapper down there? There's also, of course, a crinoid.
That's a small sea bass.
Sea bass. Okay.
You know, this is a small sea bass in the genus Plectrantheus. It's a distant relative of the groupers. They used to be placed in the same family. They're separated out now. So it's not a snapper. A little sea bass.
Before you leave here, if you could zoom in on that red urchin that was off to the left a little bit, pretty close.
Copy that, Chris.
I could swear that I saw the spines were dividing on it, but maybe it was an optical illusion.
All right, this will be our last look, folks. And Chris, can you confirm you think that was, what was it, Uchisamia?
Uchisamia.
Yeah.
It could be, I don't know, it's dendrophyllid, I think.
Is this the urchin you were talking about?
Yeah, so this is probably in the genus Histosideris. This is another pencil urchin in Siderity. And now that I can see it, I apologize. These spines aren't dividing. They were, at least I don't think they are.
I don't see any dividing spines. Yep. They're all separated.
Yeah, so these are a type of urchins that we suspect are coral eaters. And so they're almost as evil as some of Chris Maugh's sea stars.
Yeah. We saw one earlier, Del. Do you remember?
Yeah.
Was it on a coral? Or was it just
I think so.
Yeah.
Yeah.
Okay. All right, pilot. Thank you.
So for our shore-based scientist, we're going to do the dive planning call in just a couple minutes here. We'll get our mapping lead back in here. So please hang on just a moment later. And everyone else, enjoy the water column. We are planning a dive tomorrow up in Kingman Reef. So tune back in. And again, we'll have our dive planning call for that dive tomorrow in just a couple seconds.